All right. So, where do we begin with our lecture? Hang on. Okay. May nag-type. Saglit.
Sir,
Yes po. Saglit.
sir,
Chill.
okay po.
Dali, chill ka lang. Well, wait lang. Chill ka lang, okay? Chill ka lang. Ito na, isi-send ko na yung link. Sorry, wait lang. Okay, I'm here na.
Demanding pa yung
Demanding
assistant.
yung bago nating ano. Medyo okay pa yung isa dati, no? Medyo mukhang, may pagkamukhang ano lang yon, 'di ba? Ganon. Ayoko magsalita. Ganon. Alright. So, ito na, ha? Ito na. Okay, ito na yung link. I'll type it here na lang. Okay. So, here you go. Mark, you just check it out. Check it if it's okay. And you guys can also subscribe if you want. You have access to everything. Okay. You have access to everything. Everything there is free. You can watch my videos there. And that's specifically for this lecture. That link over there is specifically for this lecture. Okay. So, now that we're done with the technical side of my lecture, we're going to go to the academic side of my lecture. Bacteriology basics or bacterial basics. So we're going to look at this bacterial cell for a while first. Okay? So one of the things that we talk about in your studies, especially in the beginning of your studies in microbiology, even I think in biology 1, is that you talk about the functions of the bacterial cell wall or the components of the bacterial cell in general. Okay? So question 1 is we're going to start with what are the functions of a bacterial cell wall? Okay? So there's two things. When I put a number on an open and closed parenthesis, it means that I'm asking for two things. So the two things that you need to remember when you're talking about bacterial cell function is um, cell wall function is that it protects the internal structure of the bacteria and it provides the shape of bacteria. Okay, so now we're talking about shape of bacteria, by the way, and what are the examples of the shapes of the bacteria that we're going to study? The general shapes of bacteria. Anybody in our chat, unmute yourself. Give me an example of a shape of bacteria. It could be one. It could be all three if you like. Okay, one, two, or all three. Alexis Villabona.
Rod shape, sir.
Okay, another one. Another one. Diba DJ Khalid? Another one. Ganern. Who else can answer? What about Arnie Salan? He's looking pretty gloomy there. Um, Arnie, what other shape of bacteria can you give me? Another example, aside from rod shape.
S- Spherical po.
Spherical. Very good. What else is there? What about Crisia cineris? Crisia. Shape. We're talking about shape. So we have rods, we also have spheres. Okay? What about you, Crisia?
Ano po, corkscrew shape po.
Corkscrew shape? I want it to be a little bit more specific. Can we give a little bit something a little bit more specific? Thank you for trying, Krisha. What else is there? So we have rods, we have spheres, we have what else? Nakalimutan nyo na. What about Oh, somebody already raised their hand. What about Eliza? Eliza or Eliza? Eliza? Eliza. Naboboard ang ano natin. Naboboard ang ano natin, oh. Ang ating what do you call this? Moderator. Sabi niya,
Sorry po.
all right, who else here? Shape of bacteria. General question. Well, guys, this is basic. Angelica Grace. Sige, Angelica Grace.
Koksay po.
Yes, she said already, spheres. Okay, what else? Aside from spears, rods, and what else is there? There's three only. Marivic Sastre. Marivic Sastre.
Helical.
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 Helical. Um, rods can also be shaped as helical. Okay, try again. Thank you for participating. Don't worry, I will not judge you for any mistakes. We're here to correct it. Okay, what about Joy Lee Poras?
Spiral po.
Very good. Okay, so those are the three things. And if we want to be specific, we're going to use the nomenclature given by scientists before us. And yes, I am considering you guys as scientists. 'Di ba? Alam ko mapapangiti kayo. 'Di ba? You guys are considered scientists na, 'di ba? Kiniklaim ko na napapasa kayo, ha? Ewan ko lang dito kay Sir Marco kung i-claim niya ipapasa kayo, pero ako kiniklaim ko na napapasa kayo. So, the nomenclature given to them is usually cocci, bacilli, and spirochetes. Okay? So, cocci, bacilli, and spirochetes. So, general shapes lang yun. Tinatanong ko muna. Okay? Next. So, example of components that you will see in the bacterial cell wall. We have M protein and the mycolic acid. Okay? M protein usually prevents bacterial phagocytosis. Usually it's actually one of the pathogenic determinants of Streptococcus pyogenes, for your information. We're going to speak about it later on when we're talking about specific microorganisms, but we're still in the early stages of our discussion, but we'll discuss it later on. But just for your information, the function of the cell wall, some of the components could have M protein, and it prevents bacterial phagocytosis. Another one is mycolic acid, and it prevents intracellular digestion, usually seen in species of Mycobacterium. Okay? We'll have a specific lecture for these ones, but for the purpose of this discussion, you are coming in this discussion with the assumption that you've already known these organisms. You have known them in your third years, I suppose. I'm not sure if the curriculum has changed in the Philippines ever since I was there, but you are coming into this discussion knowing who the microorganisms I'm talking about right now. So if you're not familiar with them, I would suggest you go back, read it, come back again in a later time, and that's the reason why I always record my lectures with you guys. Okay? All right? Clear? Clear?
Clear po.
All right. So in some books, it prevents sulfonamide digestion for mycolic acid. I forgot to mention that one because that has a little bit bearing in the discussion later on. Okay, now, which species of bacteria lacks a cell wall in the structure of their cytological structure? Okay, so we have what? Who can answer this one? They lack a cell wall structure in their cytology. There's two organisms that are known to have no cell wall in their bacterial cytology. Who is that? Somebody raise their hand. Valant Acenas. How do I read your name? It's Valiant. How?
Valiant po, sir.
I love it. I love the voice. I love it. Somebody looks like Minty Fresh. I love it. Natutuwa talaga ako, Marco, sa mga set ng students natin ngayon. They're very pretty. All right, Minty Fresh. I'm calling you Minty Fresh right now. All right, now, Minty Fresh, what are these microorganisms? I love it. Smart na, pretty pa. Gusto ko yung mga ganyan.
Mycoplasma and urea something. Sorry.
It's okay. Girl, it's okay. Girl, I love it. The fact that you're answering is already a bonus for me. Minty Fresh is correct. Okay? So I dub the Minty Fresh. Today's lecture, you're one of my favorites. Okay? All right. So Minty Fresh is right. Mycoplasma ureolyticum, if you want to be Mycoplasma pneumoniae, if you want to be specific, and another one is Ureoplasma ureolyticum. Okay? By the way, these organisms are actually these are organisms that you would be discussing in future lectures, especially if you're going to talk about cold agglutinins. Okay, so please be mindful of them. So it's not only important in our discussion in microbiology, but it's also going to be important in your discussions in blood banking and also in immunology and serology. So be mindful of these things. So if I were you and you got this, I'd like you to write in a piece of paper that you have there with you, put a star on this one, or write number 3. Sabi ni Sir Manuel, important siya, because this one is really important. Okay, it's a high yield question, if I may say so myself. What else is there? Because of the absence of its cells, the structure of these organisms are termed as what? So, sige, sino makakasagot nun? Because of the absence of its cells, or cell walls, supposed to be apostrophe S, okay, of its cell walls, the structure of these organisms are termed as what? Wag na nating pahirapan yung mga estudyante. Sige na nga. We're going to call it pleomorphic, which means it's malleable, it varies in shape depending on how you fixed it or when you fixed it. Parang, it's like when you take a photograph, nag-post si Minty Fresh. Gumanyan si Minty Fresh, di ba? Nag-post siya. Hindi na siya gumalaw nun. That's the time that you captured it. So, imagine in each slide that you're preparing, I'm not sure if you guys already made experiments on preparing bacterial suspensions because of the pandemic, okay? So, I'll explain to you guys how fixation works. So, what happens in fixation is you're basically stopping time, okay? So at the moment you've fixed a particular organism in the slide, it's basically a snapshot of what they look like. So it means that if it varies in shape, it doesn't necessarily mean that that's the only thing that you'll see. That's the only shape you would see it. You would see the organism. For example, you allowed the organism like mycoplasma to grow in a particular media, and you fixed it. Okay, you fixed it in the slide. Wait, hang on a sec. Let me use a highlighter, a laser pointer highlighter. Let me, hang on a sec. How do I open my paint? Paint. So for example, can you guys see it in my screen? I'm not sure if you can see it. Can you guys see it in my screen?
Yes, we do po.
Yeah. So I have paint in my screen, right? So for example, this is your organisms. Normally you would see it like this. Okay? Sorry. Normally, you would see the organisms. Because for example, oh gosh. What am I doing? What the heck am I doing? Hang on a sec. So, there you go. So you have organisms that look like that. Okay? Now, suppose that Minty Fresh is a colleague of mine. She's inexperienced yet. She's inexperienced. She just got out of graduation, and the process at which she makes her smears are different than mine. And she got organisms that look like this. But it came from the same colony. All right? It came from the same colony. That's what you call pleomorphism. Okay, so it depends on how you stop time the moment you fix the organism in a slide. Okay? So that's known as pleomorphism. So I want you guys to note that because I'll ask that in a question. Maybe I'll ask that in a question kasi sabi ni Sir Marco, pahirapan ko daw kayo. Kasi parang gumagaling daw mga students ngayon. So, papahirapan ko daw sila pag gumawa ako ng exam. Once a week, I'll make 5 questions kasi matagal ko silang students, no Marco, hanggang November. So once a week for the rest of those 3 months, I'll think of 3 questions. Once a week lang, sir. For 1 week, I'll think of only 3 questions and I'll make it as difficult as possible. Diba? Because I want you guys to be prepared. You guys don't have a lot of face-to-face action as opposed to your previous seniors rather. Okay? So I want you guys to be to have as much laboratory experience as we can on what do you call this? An asynchronous lectures. So as much as possible. Actually, gusto ko talaga kayong masama sa laboratory para at least kahit papaano, 'di ba? 'Di ba? Gusto niyo 'yon? Gusto niyo 'yon? Gusto ko talaga si Minty Fresh. Siya lang yung gumaganon. I love it. Favorite ko na talaga siya. Paltan na nga natin yan si Mark, hindi siya nagre-react. Parang gusto ko maging moderator ko si Minty Fresh. Charing lang.
Okay lang naman
Hindi
po,
na
sir.
hindi na pwede touch move na ako. Baka sabihin I don't have what you call this?
May favoritism.
Oo.
Magwewelga ako.
Oo, ganon. Bakit? Ayaw mo bigyan ng passing grade yan si Minty Fresh? Alright.
Hindi naman, but nagpresenta na kasi si Segi.
Sorry na. O yan na, baka sabihin wala akong one word. Ano ito? Tawag doon? Wala akong sense of honor. O diba, ganun. Okay, now, main constituents of the bacterial cell wall. I think you guys already know this, but let me just ask you guys, what are the main constituents of the bacterial cell wall? Okay. So we have peptidoglycan. Okay. And the main constituent of it are the following chemicals. So if you guys remember your biochemistry classes, you'll be safe in this question. Okay? N-acetylglucosamine and N-acetylmuramic acid. Okay? So those are the main components of your peptidoglycan layer. Okay? Peptido, meaning there is protein or something nitrogenous in it. Glycan, meaning there is some sort of sugar component in it. Okay? So N-acetyl, it's already nitrogenous by the name itself. Muramic acid, okay? It's already a glucose. Glucosamine, okay? So it's all peptidoglycan, so protein. It's protein glucose combination. So if you guys remember your basic biochemistry, you'll be safe in that kind of question. Okay? If I ask bacterial physiology. All right? Now, what is the test used in differentiating bacteria based on the components of their cell wall? This is again one of those questions. Guys, this is one of those questions that you must remember. Otherwise, you got to go back to your third year classes if you don't know how to differentiate bacteria based on the components of their cell wall. So who can answer that one for me? First bullet. Anybody? Alright. Oh, taray talaga. Bet ko talaga pag smart na pretty pa. Sige, minty fresh.
Gram stain.
Very good. Gram stain. Now, another person. Ayoko nang may favoritism kasi baka magalit sa akin si Sir Marco ninyo. Sige. Who else can answer the second bullet? Indicate the reagents and their purpose in chronological order. I want it in chronological order. Nangangati na si Valiant. Gusto na niyang sagutin. Sino? Sino makakapagsagot? Ay, walang sasagot. Sige. Ang hindi makasagot nito, babagsak sa next test ni Sir Marco. Sige. Wala talaga. Walang gustong
Sir,
mag-
sa'yo galing yung exam.
Kaya nga eh. Syempre, kaya babagsak sila kasi hindi nila masagot. Oh, bilis. Who can answer? Oh my God, Deandra! Deandra, hello!
Hi, sir.
Oh my gosh, it's been such a long time, Deandra. Alright, now Deandra, can you answer the question? Indicate the reagents and their purpose.
Crystal violet po for primary stain.
Uh-huh.
Grams iodine, more than.
Very good.
95% ethyl alcohol. Decolorizer po.
Alright.
And safranin for counter stain po.
Okay, very good. Love it. Love it. Bigyan ng jacket. I love it. Okay. So I love the fact that she was actually specific with the type of decolorizer. 95% ethyl alcohol. You could actually use acetone alcohol, but that depends on it depends on the manufacturer. Sometimes they would give you 95% alcohol, sometimes it's acetone alcohol. So depending on the manufacturer, that's what you're going to get. But please don't forget acetone alcohol, okay? 95 ethyl alcohol is wonderful, but acetone alcohol is most commonly used. All right? Very good. Now, given that you already know the components of the gram stain reagent, indicate the reaction for something that is gram positive. An organism that's gram positive would have what type of color? Sige, gram positive. Nako. Mark, Segi.
Sir, purple po.
Okay, very good. Oh, 'di ba bright na pretty pa? 'Di ba bright na pretty pa? I love it, 'di ba? Very very good my moderator. I love it. Okay, what about for gram negative organisms? Sige. Basics. We're going with the basics, of course. Joyliporus.
Pink po.
Pink. What's another color? Because in
Pink or red.
Okay, red. Okay. Another one you might have forgotten is salmon red. 'Di ba yung mga ganon? Alam ko maarte ang yung mga books natin. May salmon red pa, 'di ba? Salmon pink, mga ganon. Please don't confuse those. They could be used synonymously in your exams. Okay? So they might give you a case card that says or a case presentation. Pus sample was submitted to the laboratory upon gram staining, salmon pink colonies, upon gram stain of colonies, salmon pink rod-shaped microorganisms were seen. So basically that means that it is gram positive. Sometimes they wouldn't say it's gram positive. That's why I want you guys to be familiarized with the basics before we move on to actual case presentations. Because I will be asking case presentations when we talk about organisms in specific. Okay? Cool? All right? So alam ko medyo boring. I know some of you already know some of this, but it's going to get tougher than this. And if you need assistance, you can raise your hand, you can ask me questions, or you can send a message there, and if Mark sees it, you can tell me, Mark. Okay? The only one who's allowed to speak without me asking the question is actually Mark, because I don't like sudden interruptions. Mas maganda yung meron tayong form. And that's the reason why I always ask for a moderator. Not only to look at my live stream, but also to put a little bit of order in our discussions. Okay? So, next thing that we're going to do is we're going to talk about the most common methods of fixing an organism or a specimen. Not fixating. I started making this when I was in college. I'm terribly sorry. So my grammar seems to have lost its way back then. I wasn't listening listening to English. So please, it's fixing a specimen on the slide. So let me see if people actually remember their laboratory lectures. What's the most common methods? What are the most common methods? Give me one, just one. Anybody here? Anybody? Fixing a specimen on a slide. Okay, Nigel?
Heat fixation po.
Very good. Another one. DJ Khaled. Another one. Glenn Elian.
Methanol fixation.
Methanol fixation is for blood. Okay, that's for blood. But thank you for participating. Don't worry. It doesn't mean you don't know anything. It means that you are willing to learn. Okay? So wag kang mag-alala. I don't hold that against you. Wala pang grade to. Okay? So, what's another one? Another one we can use is slide warmers. Diba? Ang arte, sir, bakit ganon? Heat fixation to slide warmer. Ano yarn? Ano bang ginagawa natin sa heat fixation? Yung may apoy, diba? Aapoyan mo lang. Diba? Tapos padadaanin mo doon. In very affluent hospitals, we have slide warmers. So, pag susyala ang lab natin, diba? Todo na to. Gumagamit tayo ng slide warmers kapag may budget, hindi kinukurakot ng PhilHealth. Oops! Sorry! Medyo political. Saka na lang kapag medtech laws ang topic. Pero let's go na to another topic. The most critical step in gram staining. This is actually a question not only in ASCP recall questions, it's also seen in many exams that you might be taking in the future. If you want to go to the Middle East, you're going to see also questions like this pop up on the exams. The most critical step in gram staining. High yield again. Because 95, it's a must know. Who knows the most critical step? Most critical step in gram staining. Glenn Elian ulit. Ayan. Revenge of Glenn. The Revenge of Glenn. Sige.
I'm not sure. Decolorizing.
Very good. You're actually okay. That's good. I like the honesty. Thank you so much, Glenn. Alright. Parang hindi na sumasagot si Minty Fresh. Kaya, nako. Parang ang favorite ko na si Glenn na. Paiba-iba ang favorite ko ngayon. Diba? Kanina si Mark kasi siya yung unang-unang nagsalita. Ngayon si Glenn na. Alright, now Glenn, this is a component of the cell wall that binds to magnesium. Okay? So it's a component of the cell wall that binds to magnesium. We're going to talk about magnesium later. Okay? Okay? The importance of magnesium is quite relevant to our discussion. That's why I want to talk about it in terms of bacterial physiology. It's actually teichoic acid. Okay? Teichoic acid. Not teichoic, it's teichoic acid only. Teichoic. Walang I ha. Teichoic acid. There's a difference. You'll know later. Okay? Now, the structure of gram-negative bacteria is different because of what? Because of gram-negative bacteria, they have a lipopolysaccharide bilayer. Okay? And sometimes they can be known as an O antigen. It could also be known as a core antigen or lipid A. Okay? Lipid A. Whilst for gram-positive bacteria, this is absent. Okay? It's absent in gram-positive bacteria. But what gram-positive bacteria have is teichoic acid. Not teichoic, ha? Teichoic acid and lipoteichoic acid. Okay? So that's the difference between the two. Okay? Present in gram-negative bacteria but absent in gram-negative. Okay? So good. So we got that clear? All right. Now, this is something that has been passed from previous generations of medtechs reviewing. This is a cue card that I usually teach my students because there are questions. All of the following are gram positive except, and then sometimes they would forget. My gosh, 'di ba? Sayang ang points, 'di ba? Sayang ang points. So I'm passing the baton to you guys. Okay, you are the youngest generations of medtechs who are graduating. Talagang kiniklaim ko na who are graduating. Kailan ba ang graduation nila? Sir Marco, 2023 ba or 2030? Kasi walang active. Charing. 2030. Ang sama. Ang sama. Wag naman. 2023. Tama ba? 2023 ang expected ninyo? Tama ba, Mark?
Tama ba? 23 kasi they're supposed to be interns na.
If they pass the exams, of course, it is conditional, of course.
Hindi
But-
sir, because of the new curriculum, sabay na yung internship and seminar 1.
Totoo ba?
Yes po.
Oh. Ang sad naman, hindi nyo ma-enjoy ang internship kasi papasok-pasok kayo sa ospital, ay sa school.
Hindi sir, we have interns na
Aha. Tapos?
na pinapapasok sa ospital.
Okay. Talaga?
Ay, sila postigo.
Okay. Hindi na pala katulad dati na wala nang school. Walang ganon, walang school. Pag intern ka, seminar lang ang papasukan mo. Meron na pala yung kasama na sa kasama na sila, ganon.
Mm.
Okay. Ang hirap naman ng buhay. Mahirap makahanap ng jowa sa mga ganyan na mga scenarios. Paano ka makakahanap ng jowa niyan? Minty fresh. 'Di ba? Mahirap, lalo na si Nigel. She's very pretty pa naman. How can you find the jowa there? Like, of course, you're going to mingle with other medtechs, 'di ba? And that kind of curriculum kind of sucks. But let's move on. Okay? So I'm going to pass the baton to you guys. Dinadagdagan ko siya every year. Lagi kong tinitingnan kung may relevance pa ba tong slide na to. And this is one of the slides that I am proud to say is proudly mine. Okay? From my previous professor. He always tells us, all keep on learning. Okay? So, ito lang yung keep on learning ko. Itong slide lang na to. Ito actually one of the 2 slides lang to. So, sometimes kasi inaalis nila, sometimes may dinadagdag sila, sometimes binabago yung pangalan. So, yun. All gram-positive cocci of medical importance dapat to. Okay? All cocci are gram-positive except for. Sige, magbigay kayo ng 3 na gram-negative that we will discuss here. Clarice Salabo.
Neisseria po.
Okay, another one. Sige pa, gusto ko ikaw na. Itodo mo na lahat to. Neisseria, okay.
Vilonella.
Okay, pronunciation is a little bit messy, but whatever. Let's go. Okay.
And Branhamella po.
Okay. What's another name for Branhamella?
Ay, nakalimutan ko na po, sorry.
Okay, so who knows
Hoy,
the other
Salabo.
name? Nako, Branhamella is no longer the name for that organism, unfortunately. So we need to update, okay? We need to update our notes, okay? So who knows the answer? What's the current name or current nomenclature? Maria Cristina something. It's Kai something. Yeah, Maria Cristina, what's her name? What's her face?
Christina po.
Okay,
Moraxella.
All right, Moraxilla. Okay, very good. Thank you so much. Unfortunately, thank you for helping out Clarice. Okay, and thank you for participating. You're very pretty as well. You're pretty as me. You're as pretty as me. I love it. Okay, let's move on. Let's move on. So very good. All of the following exhibit red fluorescence. Sorry, which of the three, rather, that we've discussed exhibit a reddish fluorescence when exposed to UV light? This is not really a must-know question, but rather it was a question that was asked in the PAMET quiz show. So if we have contenders here, you might learn a thing or two from this particular slide. Who among the three, Villonella parvula, Neisseria species, or Moraxella species? Moraxella cataralis in particular. Villonella parvula, Neisseria species, Moraxella cataralis. Who among those three, or which among those three rather, who, I'm referring to them as if they're a person. Diandra, sige, the chosen one.
Villonella po.
Very good. Villonella ano? Huwag mo akong chinacharot. Alam mo, ayoko ng hindi specific. Ano? Diandra.
Isa lang yan. Isa lang yan.
Diandra, nanggigigil ako sa'yo, Diandra. Kakasabi ko lang kanina. Thank you so much, James. Bakit hindi ka nagsasalita, James? Our recitation is supposed to be microphone-based. Kung gusto nyo makakuha ng plus. Or plus ba? Or ano? Ay, hindi pala. Cash prize pala. Ayan. Bakit ka hindi nagsasalita? Ano? Oh, he's very handsome naman kala, girls eh. Bakit ayaw niya magpakita? Siguro meron tong X dito. Ayaw magpakita dito. It's quite handsome. And not in a predatory sort of way. Like a handsome kid of mine. Diba? It's not in a predatory way. Baka mamaya mai-report ako eh. It's not predatory, my goodness. Pagka mga estudyante ko na hindi ko yan tinuturing na mga ano, ha? Huwag kayong ganun. Walang issue. Diba? As a tita, I would recommend James, ha, guys. I would recommend it to you. I would recommend him to you. Alright. Alright, so Villonella parvula, the other name of Branhamella is now, the contemporary name rather, or the contemporary nomenclature of Branhamella is now Moraxella and Neisseria. Okay, Neisseria species. Villonella parvula is the one that exhibits red fluorescence in UV light. Where's the picture of that one? I have a picture of that one. I'll show it to you guys later. Anyway, medically relevant gram-positive. Examples of medically relevant gram-positive aerobes. Okay, gram-positive cocci aerobes. These are the things that we're going to talk about in our next lecture. Okay. Medically relevant gram-positive cocci aerobes. Okay, there's 3 that we're going to be discussing. Okay, sige lang, kahit isa lang. Sige na. Sige. Go. Mark. Do not disappoint me, Mark.
Wait lang po.
Wait lang po, ginugoogle ko. Alright. Medically important gram-positive cocci. Naku, magagalit si Marco sa inyo. Magagalit si Marco. It's a must know, guys. It's a must know. What about my previous favorite Minty Fresh? Parang hindi na siya nagpaparamdam. Itaas mo ang bandera natin, Minty Fresh. Hindi pwedeng ganda lang. Nasaan na si Minty? Nawawala si Minty. Nako, ha? Okay, who can answer? Who? Anybody? Narinig ko bumubulong si Glenn, pero hindi nakikita ko bumubulong si Glenn. Sige na, sabihin mo na sa akin kung ano yung binubulong mo. What are you whispering, girl? Whisper with wings. Ganun. Oh, what? Medically relevant gram-positive cocci. O, bibigyan ko kayo ng clue. Beta hemolytic, yung mga inaano natin, mga beta hemolytic. Coagulase positive, coagulase negative. Oh, wala pa rin? Doesn't ring a bell? Guys? Anybody? Gram-positive cocci. Mamaya na tayo dun sa anaerobes. Kasi parehas lang yung pangalan. Halos parehas lang yung pangalan nila eh. Sige. Parang gusto ko na magbigay ngayon ng pera. Sige, parang gusto ko mamigay ng pera ngayon. Sige, sino makakasagot? medically relevant, 3 lang sila, guys. Nagigigil ako sa iyo. Nagigigil ako sa iyo, Mark. Hindi ka sumagot. Sige. Medically relevant. Sinabi ko na ngayon, coagulase positive, beta hemolytic, grows on blood agar. Oo. Wala talaga. Siret na kayo, guys. Oh, may nagsabi. May nag-raise ng hand. Sino yung nag-raise ng hand? Ay, may nag-chat. Sino yung nag-chat? Wala naman nag-chat. Ay, ayan, James. O, sige, James. Sige.
Sir, try lang. Stop
Sige.
po, tsaka strep.
Oh, ano yung isa? Kulang? Wala?
Strep po.
Okay, stop. Strep. Strep ano? I need you to be specific. Huwag ganon. Alam nila di, Andrea, gusto ko ng complete. Staphylococcus. Okay, the other one is?
Staphylococcus po.
Okay. And then the other one is streptococcus. All right. Now the other one, what's the last one? Because it causes a lot of confusion to many scientists and medical laboratory professionals. The other one is Micrococciae, Micrococcus species. Okay? Micrococcus species. Bakit siya kailangan i-discuss, sir? Akala ko ano siya, medically relevant because it causes a lot of contamination. Okay? Blood bank sometimes has overgrowth of Micrococcus. If you collect blood culture bottles from patients who have not been properly, or rather the site of the blood collection is not wasn't sterilized prior to the collection, you might have growth of Micrococcus. So that's the reason why we always the reason being also is because of the fact that it grows on blood agar plates, and it causes a lot. Hence it is medically relevant. But we'll talk about Micrococcus in deeper detail in some other lecture. I think today or maybe next week. Okay. Now, what about gram-positive cocci anaerobes? Parehas lang yan yung pangalan nila. Peptococcus, Peptostreptococcus, and of course Sarcina species. Okay, Sarcina lutea. Okay. Specifically Sarcina lutea. Okay? Why lutea? Because it's yellow. It's the only organism that usually grows in tetrads of 4. When I say tetrads of 4, where's my paint? Where's paint? Okay? It grows in tetrads of 4. Mark, can you see my what do you call this? My paint screen here? Please tell me.
Yes po.
Okay, very good. Thank you. All right. So usually it will grow like this. If you see something like this in your slides. Tetrads of 4. That's basically what it means. Or cluster of 4. They usually appear like that. I have a picture before, but I deleted it because there's so much material here that I'm looking at. So they would appear something like this. Yeah. So when you look at it in the grime stain, it look like bricks. That's Sarcina lutea. Okay, these other two, Peptostreptococcus, basically they look like Staphylococcus. They are bunches of grapes that grow anaerobically. Okay, and Peptostreptococcus, from the name itself, it's like strep, but it grows anaerobically. Do we need to discuss them? I think this is the last time you'll be hearing Peptostreptococcus and Peptococcus in our lectures. So don't worry about that one. What I need you guys to focus on is Micrococcus, Staphylococcus, Streptococcus. That's the thing that we're going to talk about in later discussions. Okay? Now, this one is very important as well. All bacilli are gram-negative except for the 11. All right, somebody raised their hand. Oh, sige. Who was that? I did not see. Sorry. Who was that? Okay, Mark. Sige, go. For 100. For 100
Actually, hindi ko rin
Gcash.
po nakita yung nagtaas ng kamay. Hindi po ako yung nagtaas ng
Okay.
kamay.
So, akala ko pa naman ikaw, bibigyan na sana kita ng 100 Gcash. Sige. Teka lang, mag-open lang ako ng notes para alam ko kung sino bibigyan ko ng 100 na Gcash. Open ko lang yung notebook ko. Who here can answer 11? Gusto ko isang bagsakan na 11, ha?
Si Malefic
bacilli
Sastre
are...
daw po.
Okay, sige. Can I see you
No,
first?
sorry ko. Kulang po.
Isang bagsakan. Sige. Alam nyo guys, ang tanda ko na to. Matagal na ako sa sa practice. Memorize ko pa rin siya. I want you guys to have the same level of enthusiasm with microbiology as I did when I was a student. O sige, Marivic, ayan na. This is it, Marivic, 11. I need 11, okay? Nakaganyan ang kamay ko. If you're looking at me in YouTube, nakataas ang kamay ko tsaka isang daliri ko sa paa. Okay?
Ay, sir, sorry. Yung sa YouTube nyo po, wala po yung camera nyo.
Ay, nakawala yung camera. Ay, sorry naman. Okay.
Apo.
Thank you. Thank you for informing me. Alright.
Okay po.
Huwag na natin ilagay yung video ko doon. Nakakahiya. Hindi ako nagsuklay. Anyway, sige. Marvic, 11 gram-negative bacilli that we're going to talk about. Okay. Dumudugo ang ilong.
Ano
Sige,
po?
push.
Arcanobacterium, Bacillus clostridium, Corynebacterium, Erysipelotebrix, Gordonia, Cutbria, Listeria, Mycobacterium, Nocardia, Rhabdococcus, and Sucamarella.
Kulang. Actually, sobra. Pero, hindi kasama si over ka naman. Ba't kasama si Arcanobacterium?
Ay, nakalagay mo dito, sorry.
Nako,
Nakalagay mo
bakit
dito.
may Arcanobacterium? Sir Marco, bakit kasama ang Arcanobacterium? Ano ba itong mga students natin? Biology students ba itong
Super
mga
advanced sila.
Ha?
Hindi nga lumalabas sa board exam yun.
Kaya nga, sabi ko, yan, alam niyo kung ano yung mga Arcanobacterium, guys? Yun yung mga bacteria na nag-grow sa Mars. Yung galing sa mga mga Martian stones. Correct me if I'm wrong, Sir Marco, because I remember when I was studying microbiology way back, a teacher of mine said, you know, Arcanobacterium, from the name itself, arcane, which means it's old, means it's primitive. Okay, so it's probably the what do you call this, the earliest forms of the bacteria. Okay, so usually these are seen in anaerobic conditions like in Mars. So yun, so Martian stones.
Andito
Sabi
po
ko,
kasi
bakit
siya
may
sa notes ko, X ko na lang po.
Alright, so sino pang pwedeng mag-try? Gusto ko nakataas noon, nakataas ang baba. Kasi yun ang i-discuss natin. Sige, Mark,
Sir, genus
go.
lang po ba?
Oo! Bakit
Genus
kung gusto mo lang.
Pseudomonas
Hi!
alcalici.
Umpisa pa lang, X ka na. I will stop you there, sir. Sinabi mo,
Okay po.
Pseudomonas?
Pseudomonas.
Bakit may Pseudomonas? Shoot at this live.
Pseudomonas po is gram neg.
Pseudomonas is gram-neg.
Okay.
Ay, nako. Eliminated with the first set of
Okay po.
microorganisms. I'm terribly sorry. Thank you for trying.
Okay.
Alright. Who else? Sige. 2 students pa pwede mag-try. Alexis Villabona.
Try ko lang po, naaalala ko po. Ayun,
Sure.
Mycobacteria,
Okay.
Actinomyces,
Aha.
Bacillus,
Aha.
Lactobacillus,
Aha.
Listeria,
Aha.
Corynebacterium,
Aha.
Erysipelotebrix, Propionibacterium,
Aha.
Rotia, Nocardia, then yung isa po, Pertia, Actinomyces.
Kulang ng isa. Binibilang ko. Kulang ng isa. Sayang! Bilisan mo, bigain na natin. Wala kasing organization kaya nalilito kayo Mamaya pakita ko sa inyo yung organization niya. Napaka-simple lang.
Mycobacterium, Actinomyces, Bacillus, Lactobacillus, Pertia, Nocardia,
Kulang. Tiret na?
Kulang
O,
po.
sige lang. Sige, minty fresh. Ayan na talaga. Itaas ang bandera ng mga Becky. I love it!
Guys, yung kulang, napaka-importante nun.
Yeah, I know, right?
Mycobacterium.
Oh, ayan na. Sige na, minty fresh.
Mycobacterium, Bacillus, Clostridium, Cornean Bacterium, Lactobacillus, Listeria, Erysipelotrix,
Aha.
tapos Erysipi, ay hirap din kasi.
That's part
Tapos
of the challenge.
Actinomyces, Rotia, Cortia, and Nocardia.
Urtia, Rothia, Nocardia. Tingnan nga natin kung tama. Pero may makukuha ba siyang cash prize? Tingnan natin. Sandali. Nasaan ako? Kulang ka ng isa. Kulang.
Ulitin ko po. Mycobacterium, Bacillus, Clostridium, Corine Bacterium, Lactobacillus, Listeria, Erysipelotrix, Actinomyces, Rothia, Curthia, and Nocardia.
Kulang talaga. Alright. Walang nanalo. This is our answers. Organize nyo kasi. Aerobe starts with Bacillus, Corynebacterium, Erysipelotrix, Listeria, Lactobacillus, Mycobacteria, Nocardia, Rothia, Curthia, Actinomyces, Clostridium, Propionibacterium is the last one that you did not say. Propionibacterium. Okay? Yun yung wala. Meron si Alexis, wala ka naman. Sayang. No? Very strict tayo. Sayang. 100 peso Gcash na yun.
Hindi na po pinaghihiwala yung Rothia and Curthia, no?
Oo. Never natin pinaghihiwalay ang Curthia, Rothia because they belong to almost the same genetic markers.
Doon sila nahihirapan sa bilang kasi magkahiwalay.
Yes. So, Curthia, Rothia. Okay? Are just one group of organisms that are considered to be extremely close in terms of genetic familiarity. Okay? So, paano ba inayos? Paano mo inayos, sir? Anong pinagsasasabi mo, sir, na inayos? Parang magulo pa rin para sa amin. Look, by letter, para madali niyong maalala. Nakita nyo? Kung ako kahit nakapikit, memorize ko, Bacillus, Corynebacterium, Clostridium, di ba? Clostridium, Erysipelotrix, Listeria, Lactobacillus, Mycobacterium, Nocardia, Curthia, Rothia. Okay? Actinomyces, Clostridium, Corynebacterium yung kanina sinabi ko, Propionibacterium. So, ACP, di ba? Madali, di ba? ACP, di ba? And then, kayo nang bahala dun sa Bacillus. Dun sa group ng aerobes. Diba? Arrange it according to letters or according to alphabetical order para hindi kayo mahirapan. Sometimes the reason why I want you guys to remember it in according to letters is because that's also the way we're going to discuss it. Okay? We're going to start with Corynebacterium. Actually, we're going to discuss Bacillus and Clostridium together, but the rest of them we're going to discuss it according to letter. Okay? Or according to alphabetical order. Alright? So, okay na yan. Huwag kayo mag-alala. You don't need to write. Okay? You don't need to write. You're going to get a copy of this. Hindi naman ako ganun kadamot. I'm not that kind of monster. Okay? Alright. Now, let's move on. Okay? So, we have fungi or fungus-like bacteria. Who can answer this one? Looking at the list, who among those 11 organisms are fungus-like? Or they share similar characteristics to fungi? Who can answer this one? Who raised their hand? I didn't see who raised their hand. James. Sige, James.
Actinomyces po.
And the other one? Who's the other one? Okay, you have one from the anaerobes. You have one also from aerobes. Which one? Which one from Bacillus, Corynebacterium, Erysipelotrix, Listeria, Lactobacillus, Mycobacteria, Nocardia, and Curthia, Rothia is fungus-like? Maria Cristina Caye Sabado.
K po. Actinomyces and Nocardia po.
Very good. I love it. Smart na, pretty pa. I love it. Very good. I love it. You're as pretty as me. Diba? Ganun. Talagang kiniklaim ko kahit hindi mo ako kasing ganda. Kasi ako lamang. Charing! Charot. Okay. Ako lamang ang nag-iisa. Charot. Okay, so fungus-like bacteria, actinomyces, and Nocardia. So we're going to discuss actinomyces and Nocardia in a later topic, but let's be specific. Actinomyces what? And Nocardia what? Who are we going to discuss? Actinomyces Israeli and Actinomyces Israeli and Nocardia asteroides. Okay? So we're going to discuss those things. Okay? Example of spirochetes. Oh, ayan na naman. Okay? Ayan na naman. I want you guys to remember TBL. Tomboy, bakla, lesbian. Ano yan, sir? Ano yung TBL na yan? Tomboy, bakla, lesbian? Hoy, redundancy. I will never make one. Tomboy is already a lesbian. Kayo talaga ha. Huwag ganon. Okay? Example of spirochetes. Let's go. Who knows what TBL stands for? Marivic Sastre. Okay. I want to see your face also, girl.
Treponema borrelia.
Okay, ano? What?
Treponema borrelia and Leptospira.
All right, very good. Thank you so much. Now, the special term used for their flagella to associated to their flagella is known as axial filaments. Okay, or periplasma or periplasmic flagella. Okay, so axial filaments. So they have flagella also, but it's known as axial filaments or periplasmic flagella. Okay, so she's correct. Treponema pallidum is something that you need to remember. Because why? What's the causative? Treponema is the causative agent of what? Treponema pallidum. Alexis.
Syphilis, sir.
Very good. I think somebody's studying their microbiology. I love it. You're my new favorite now. Okay. Ang dami kong favorite, no? 'Cause I love you guys so much talaga. What about Borrelia burgdorferi? What does it cause? Borrelia burgdorferi. Causative agent of what? Valiant acid. So talagang hindi talaga nagpapatalo ang mga ano. Okay, raise that pride flag, girl. What is Borrelia
Lyme's
burgdorferi?
disease.
Lyme disease. Very good. Now, what causes? What is the who transmits or what transmits Lyme disease to humans? Sige. Patay. Tumba tayo, sir. Ticks or fleas. Okay? Ticks or fleas. Okay? Particularly Ixodes. Okay? So don't forget those things. Leptospira, alam niyo na yan. Pero we're going to come back to that one some other time. Okay? There's a different discussion for that one. But I want you guys to just get reacquainted with your knowledge in microbiology. Okay? So, carry nyo ba? Gora, shall we go to the next one? All right. Thank you. Okay. Now, it is known that spirochetes generally cannot be stained. However, some of them exhibit a Gram reaction. Spirochetes that have a Gram reaction are known as what? Are what? Gram positive or Gram negative? They are what? Are they Gram positive or the only answer is either Gram positive or Gram negative. So what is it? Again. Let me repeat the question. It is known that spirochetes generally cannot be stained. However, some can exhibit a Gram reaction. Spirochetes have a Gram reaction. Spirochetes that have a Gram reaction are blank. Who raised that? Valiant.
Gram negative.
Very good. Very good, very good. Oh my gosh. Somebody is already becoming my favorite again. I love it. Di ba? Gusto mo yan? Gusto ko yarn. Alright, let's move on. I love it. Okay, so spirochetes are Gram negative. Why? Because they don't have peptidoglycan. Okay? That's the only reason why. That's the only reason why they are Gram negative. Because you can't stain a spirochete also. But we try. Silver stain, by the way, is the most common way to stain them. And usually it's you have to inoculate it in animals first. I know it's going to sound disgusting, but we're talking about it in a scientific manner, okay? So please bear with me if I sound a little bit too gruesome. Okay? So the way we try to grow Leptospira They can't be they can't be grown in artificial media. Okay? You need an animal inoculation. You need to do animal inoculation techniques. And one of the most common way is to inoculate it in hamster balls. Yes, in hamster testicles or rabbit testicles. Because they love it there. Okay? They love to grow in these places. So I know it sounds a little bit yuck, sir, but that's the course that you chose, the disgusting ones. Okay? So you need to have testicles, specifically from animals like rabbits or hamsters. Animal inoculation techniques. Okay? To actually visualize these organisms. Okay? Now, the term given to the Gram-stain reaction of mycobacteria. The term given to the Gram-stain reaction of mycobacteria. Meron ba, sir? Akala ko AFB ang ginagamit. Meron. Sige, who can answer? Who can answer? Gram what? Not gram positive or gram negative. Although we said they're gram positive, right? They are gram positive. Although we said that they're gram negative, but they're not. They have a specific term for them. These organisms have a specific term. Nigel.
Teka lang po, acid fast.
No, I just said acid fast. We're talking about gram stain first. Chill ka lang. Mamaya na yung acid fast eh. Ano yung tawag sa gram? Marivic.
Gram neutral.
Gram neutral. Very good. It's a very long time since I heard that one. Thank you so much. It's a long time since I've heard that one. Well, this question is actually a question that I took from the PAMET quiz show. They call it gram ghost or gram neutral. Okay? So we have in hematology, we have ghost cells. So here we also have ghost cells, but in this case it's known as gram neutral. Okay? So it's a PAMET question. I know there's a rare opportunity that you might be asked this question in your board exams, but it's nice to know, right? Maybe you would be asked this question. So what is the gram stain reaction of mycobacteria? Gram negative. Okay? Okay? Cool? All bacilli are gram negative except for all bacilli are gram negative. All bacilli gram positive. Sorry. Nalilito na din ako. All bacilli are gram negative except for these 11 organisms here. Okay? So Pseudomonas, yung mga sinabi nila E. coli, Edwardsia tarda, what else? Pleishiomonas, yung mga ganon. Ano yon? Gram negative yon. So, kailangan nyo itong tandaan. Because you might be asked the question, which of the following is gram positive? Diba? Kaya I want you guys to remember these 11 organisms. Okay? Cool? Kung meron man kayong i-highlight or ilalagay sa notebook nyo na tatandaan yung number, it's number 13. Lucky number 13. Okay? Alright? Next. Saan na tayo? Gram ghost. Aside from mycobacteria, which organism can be considered as acid fast or acid partial fast or partially acid fast? O, ayan na. Mycobacteria. Ayan na, Nigel. Ayan na. Alam ko, ready ka na. We're going to talk about acid fast staining na. Ayan na. Sige na. Kating-kati ka na kanina. Sige. Which other organism?
Nocardia po.
Nocardia, okay. There's 3. My gosh, sir, hindi ako ready. Nocardia lang ang naalala ko. Hindi ako ready. Alright, I wasn't ready. I wasn't prepared. Stop it, sir. But there's 3. Fungus-like bacteria, actinomyces. O, yan na, tapos na. O, di ba? May isa pa. Legionella micdadeae. Sir, akala ko ba hindi siya gram positive? Sabi ko, hoy, AFB na ang pinag-uusapan natin. Kaya gusto ko siniseparate ninyo yung mga discussions. Di ba? Kaya kanina, di ba, hoy, Nigel, stop lang tayo dyan muna, ha? Mamaya na yung AFB. Kaya kita in-stop kanina. Rhodococcus pala, sorry. Rhodococcus, isa. And then actinomyces species. Kasi under si Nocardia as actinomyces ng Nocardia. Okay? So, Rhodococcus and then Legionella micdadeae. So, Rhodococcus is more or less a family. Okay? Yeah. So Legionella make the day. Okay? So okay ba? So what is the gram stain reaction of Legionella make the day? It is not one of the 11, so therefore it is gram negative. 'Di ba? Para madaling maalala. 'Di ba? 'Di ba? Okay. Now, parasites that are known to be acid fast. Ano ba yan, sir? Bakit pati ito meron ding acid fast staining? Yes, we need to remember these because I'll also be discussing parasitology with you guys. So, I want you guys to remember these things. So, parasites that are known to be acid fast, Isospora belli and Cryptosporidium parvum. Okay? So, Isospora and Cryptosporidium parvum. Isospora belli, Cryptosporidium parvum. Okay? So, general lang to. Kasi I have a different discussion for these things. But, for the sake of this discussion, the reason we're going to touch up on it. This is usually found in the bronchoalveolar lavage of patients with with fulminant HIV or AIDS. Acquired immunodeficiency syndromes. All right. So usually they're found in either the sputum or in the bronchoalveolar lavage or BAL. All right. So alam niyo ba kung ano yung bronchoalveolar lavage? Are you guys familiar with what it is? Right? So if you guys are not familiar with it, I'll draw something. I hope it looks pretty. Hang on a sec. Paint ulit tayo. I'm not going to be discussing analysis of urine and body fluids, but basically, Mark, can we see paint in my? Can you guys see paint?
Not yet pa po. Ayan po.
Okay. So
Okay na ba?
usually when we talk about bronchoalveolar lavage, where is that one? Where is that? All right. So this is you. This is going to be you. For example, this is Nigella or Nigel. All right. So Nigel is a person suffering from pneumonia. And we want to know what's causing it because we already took sputum samples and we can't possibly find what organism is ailing her. Right? We also tested for autoimmune diseases, but nothing seems to come up in her diagnosis. So, what the doctors do is they do a bronchoalveolar lavage, a deeper suction of her lungs. So what do we do there? So that's her lungs. She kind of looks like a frog here. I'm sorry for my drawing. Okay? She kind of looks like a frog, so I'm terribly sorry. I apologize in advance. I'm not very good at digital drawing. What they do is they insert a tube that is directed to the trachea, and this one is going to be flushed with what chemical? Who here knows? Normal. Who can say? Normal. Nobody? What do we do? Normal. Sorry. Normal. What do we put to get bronchoalveolar lavage? Normal. Alexis?
Normal saline.
Okay,
And
normal
this is
saline. I love it. Normal saline. Okay? Normal saline solution. Kulang ka ng S. Solution. Okay? Pero okay lang. I accept it. Because lower saline solution. So what they do is you flush it. You flush the patient with it. And then you suction it. And then in the laboratory, what we do is we centrifuge it. Actually, we cytocentrifuge it in the laboratory to get a specific yield of this one. And then throw out the supernatant. And then we stain the remaining ones. So, Alexis, if you are, sorry, Nigel, this is Nigel pala. Sorry, Nigel, I'm terribly sorry. You look like a frog in my drawing. This isn't my best work. I'm terribly sorry. I apologize, okay? All right, this is an inaccurate depiction of Nigel. I'm terribly sorry. I apologize. Okay, I could not say sorry for him. I could not be more sorry. Okay, so what we do with Nigel is we take the sample from her. They bring it to the laboratory. This isn't done by medtechs. Okay, this is done by physicians. Okay, that's why very little is discussed to you guys. What they do is you get 2 tubes of this one. My tube is in a box. Sorry. So they get 2 tubes. One is the first drain, or what they call the first flush. Second one is the second flush. And usually what we keep in the laboratory is the second flush. Okay? Now the second flush, we're going to put it in the centrifuge. Usually it's going to be colored like this. And then when you centrifuge it, you'll only get something like this. Supernatant, something like this. You throw out the excess, you make a stain from that one, and that is your bronchoalveolar lavage preparation. Now you do whatever you want with it. You cannot culture this one, of course, because this one is contaminated already. Why is it contaminated, sir? Because you are using the solution comes from different parts of the body, right? It might be contaminated. We can't use it for culture. Very rarely is it used for culture, unless you're using selective media. Okay? But that's another story. All right? So that's the basic discussion on bronchoalveolar lavage, and that's something that you might think that you might want to ponder on in your free time. Wala lang, kung trip mo lang, wala kang magawa. Paano nga ba ulit ginagawa kumukuha ng bronchoalveolar lavage? Pag nagmumuni-muni ka sa bahay nyo, di ba James? Wala kang ibang magawa. Titingin ka lang doon. Paano nga ulit kumukuha ng bronchoalveolar lavage? Ganon. Ganon dapat pag magme-medtech, ha? Hindi pwede yung, ano kayang bibilhin ko para matuwa sa akin yung nililigawan ko? Wag ganon, James. Okay? Dapat dinadaan din natin sa sa smarts natin, James, ha? Okay? Huwag kang ngingisi-ngisi, Mark. Mamaya kita pagtitripan. Iba muna. Okay? Alright. Next. All gram-negative bacilli are aerobes except for what? Ayan na. All gram-negative bacilli are aerobes except for what? Kanina gram-positive pinag-usapan natin yung 11. Ngayon naman gram-negative bacilli naman. Sige. All gram-negative bacilli are aerobes except for what? Tandaan nyo, pag sinabi kong aerobe, pag sinabi kong except, ibig sabihin, opposite of what I was asking for. And this is usually a trick I do to students to confuse them. Ang haba-haba ng case, tapos may nakalagay, except, all of the following organisms are possible sources of pathogenesis, of the possible sources or possible origins of the disease process, except, ang haba-haba, imagine mo parang half ng one whole sheet of paper ninyo yung itatanong ko. Tapos may nakalagay sa dulo, except kung ano-ano nang inanalyze ninyo, di ba? Naglagay pa kayo ng table kung paano yung test differentiation. Di ba? Naglagay ka pa TSI, tapos may nakalagay sa babat, except, di ba? So minsan yan ang ginagawa ko, nililito ko yung mga students. Sige. There are 2. Who can answer? Parang gusto ko mamigay ng pera. Sir Marco, parang gusto ko mamigay ng pera kasi hindi sila attentive na. Natahimik na ulit sila. Nigel.
Sampulan. Mamigay ka na ng pera para
Parang gusto ko mamigay ng pera. Oo.
Guys, mayaman yan. So, sige lang. Hingi lang kayo.
Hingi lang. Tandaan nyo. Baka makakuha din kayo ng plus dahil si Sir Marco ang prof nyo, 'di ba? Malay nyo, 'di ba? Magbulong ako, "Hoy, bigyan mo naman ng plus." Pero hindi yan nakikinig sa akin eh. Char. Sabi, "Hoy, bigyan mo naman ng plus." Kawawa naman. Ganon. Mamimigay ako ng pera. Namigay ako ng pera ata kay sino ba yung kaklase ni Leandra na binigyan ko ng pera nun? Si ano ata 'yon? Basta nakalimutan ko na kasi what's her face na sila pagka na-receive ko na yung stipend ko. Bilis na. All gram-negative. By the way, when I'm asking this one, it's not not in every microbiology book, ha? Because what I'm asking is particularly focused on what exams you're going to take. Okay? It's not applicable if you're studying more advanced microbiology. It's not applicable there. What I'm focusing on is what they usually ask you in your exams. Kaya wag kayong mag-alala. Pinadadali ko buhay ninyo. Kasi kanina may mga nagsasabi ng mga microorganisms na kakaiba eh, mga arcanobacterium eh. Wag ganon. Okay? Sige na, sino nagtaas ng kamay? Hindi ko nakita. Nyaw-nyaw kasi ako ng nyaw-nyaw. Who? 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 Hindi ko nakita. Sino yung ayan,
Si
James,
James
Paul. Alright, James Paul.
Sir, trifold po. Neisseria po, tsaka Pseudomonas.
Sandali. Hoy. Sandali. Bakit naglagay ka ng gram-positive cocci? Kakausap lang natin kanina, 'di ba? 'Di ba?
Neisseria.
Neisseria is a cocci. Basila yung tanong. O, yan na. Nadali ng trap si James Paul. Nadali ka ng trap. Basilay. Oh,
Master
bigyan ko kayo
pass.
na Diandra. Sige, thank you James for participating. Okay, Diandra, sige. The chosen one.
Sir,
Hoy, sandali.
bacteroides.
Wait lang. Bakit hindi ka nagpapakita sa akin? Na-miss kita, Diandra. Hindi ka nagpapakita sa akin. Anong kaguluhan to? Ayan. Oh my
Hi,
God.
sir.
My God, she's one of my students before. She's as pretty as me. But not equally pretty. As pretty lang. Somewhat pretty as me. Ganon. Ganon ang descriptors. Sige, ano yon?
Bacteroides po.
Bacteroides. Okay. Another one. DJ Kalimulid. Another one.
Fusobacterium.
Very good. Medyo mali yung pagka-pronounce, but whatever, I don't really care. As long as you said it correctly, it's actually Bacteroides and Fusobacterium. Thank you so much. O, diba? Now, saan natin usually nakukuha ang mga anaerobes na mga gram-negative bacilli? Who can answer? What type of samples ba? Sige. What type of samples? Internal organs. Specifically, organs that are suffering from necrosis, where oxygen is depleted. Kaya nga anaerobes, di ba? Yung mga surgical wounds, mga ganon, 'di ba? Kasi nasaraduhan na, wala nang pumapasok na na hangin, and then hindi din proper ang circulation kasi there's several clots in the microvasculature, 'di ba? So you guys have to remember, dapat ganon. Alam ko mahirap, Mark. Mahirap. Napapatingin ka sa sa taas, pero I know kaya mo 'yan. Share this lucky mangga. Di ba? Ganon. Share this lucky mangga. You will pass your exams. Ganon ang pag hindi kaya ng total review and cramming, share this lucky mangga. Di ba? Ganon. Meron pumapasa na ganon, di ba, Sir Marco? May pumapasa share this lucky mangga.
Ako, last semester ata, may ano ba?
Nag-share. Yung hindi mo akalain na papasa, Sir Marco, pero pumasa. By the way, guys, disclaimer lang pala, guys. Gina-judge namin kayo ni Sir Marco after ng exams ninyo. Okay?
Naman. Alam na yung mga estudyante ko, syempre.
Oo, because I have access also to Sir Marco's, what do you call that one? Google what? Google Notes or Google
Forms.
Google Forms, yeah. So I can see the statistics. The reason why I'm also posting this in my live stream is because I want to know how many students are actually reviewing prior to their exams. Kaya nakikita ko din yung data. Okay? So alam ko kung kailan kayo nagre-review. Last year, paano nga yung statistics natin, Sir Marco, kaya ang dami bumabagsak last year? Students only view my videos 2 days before their exams. And the exams were announced a month ago.
Nagno-notify ang YouTube.
Nagno-notify ang YouTube sa akin. Sasabihin, one of your videos is trending. Ang topic, hematology. Kaya alam ko rin kung bakit kayo babagsak. Kasi, 2 days before ng exam, saka ka lang nag-review, yung content na tinuro ko, 6 hours mahigit. Diba? Imaginein mo yon, tapos, ikinondense ko pa yung sa 6 hours na yon, may mga bagay doon na kailangan tandaan mo talaga. Diba? Kaya, additional data for your professor and for your information so that future students will get a heads up kung ano ba ang ginagawa naming mali. Baka yun yung reason. Kaya ngayon, may natutunan kami ni Sir Marco na kaya pala bumabagsak ang mga students 2 days before ng exam. Tandaan mo, hematology 1 and 2. 2 days before the exam mo lang aaralin. Of course, the likelihood of you passing is not going to be high, especially when your previous performance is, of course, ang nakukuha mo lang sa exam mo, 1 out of 100 is 10. 1 out of 100 is 15. Tapos 2 hours from the exam. My God. Even a super genius will not be able to do that. Of course, Sheldon Cooper could do it, but he's a different person, of course, right? But please, please, the data that I have is enough for your professor to assess what is wrong and what should be done and what should be addressed. What can we do to address the issue? Please don't review 2 days before your exams, okay? Gawin trending ang video si Sir Manuel. Buong linggo. Para ma-monetize
Actually,
si Sir
ini-scrub lang
Manuel.
niya kayo para ma-monetize
Oo,
na siya ng
Oo,
YouTube.
para ma-monetize ako ng YouTube. Ganon! Pero hindi, totoo. Pero napansin namin ni Sir Marco yun. Di ba, Sir? Anyway.
Oo, kasi let's say exam, tapos 2 to 3 days before, mag-i-review sila ng video lectures worth around 20 to
20
25 hours
Mm-mm.
ng lecture. Malamang babagsak talaga kayo.
Oo. Imagine mo hindi na maganda ang performance mo yung 2.75 or 3 ang nakuha mo sa exam mo dati, yung final passing grade mo sa bacteriology, tapos ganun ang klase ng ng pag-aaral. No, there is something wrong with the study methodologies and we need to change it. You want to become a medtech, you want to become a doctor, you need to change the study habits. Wag 2 days before the exam, okay? Alright, na hindi ko kayo pinapagalitan, ganyan ko kayo kamahal kasi ayoko kayong bumagsak, 'di ba? Anyway, let's move on to the 2 types of AFB staining. What are the 2 types of AFB staining? Sige nga, Minty Fresh, kasi pinapatay mo na yung ilaw mo eh. Naluloka ako sa iyo eh. Sige nga, what are the 2 types of AFB staining? Bruha, naka-mute ka.
Pass muna.
Akala ko sumagot ka. Sige, who else? Sige, baka sabihin ni Mark, kinakalimutan ko na siya. Sige, Mark. What are the 2 types of AFB staining?
Sorry din po. Nag-pass din mo. Hinahanap ko pa po sa notes ko.
Sige,
Hoy, ba't kaya ganyan?
That's the most common, di ba? Yan ang kinagalit ni Sir Marco last year din. Last year yan din yung kinagalit to Sir Marco. Pwede din
Ba't kayo
naman
ganyan?
Huwag nyo na sabihin yung pangalan ng tao. Sige na. O sige, mabait na ako. Huwag nyo na sabihin yung pangalan ng tao na nag-discover. Sige, Amanda. Ngayon ko lang narinig si Amanda and she's quite sleepy. Para magising si Amanda. Alright, Amanda.
Ano po, sir? Carbon fusion po at saka...
Dai,
Ano
nato
yung
ka ba,
isa?
Dai? Dai, you're sleeping, Dai.
Sorry.
Hindi ko tinatanong reagent.
Sir,
Types lang,
chlorochrome.
te. Types lang muna
Ay, sorry.
tayo, Dai. Dai, types muna, Dai. Sabi ko na inaano.
Sorry,
I
stock
love
knowledge
it.
lang po.
Ay, okay lang yun. Sige, huwag ka
Stock knowledge
mag-alala.
lang po.
Tagay pa! Gusto ko yan. Tagay pa! Alright. Sige, go! Sige, stock knowledge. Try natin isa pa. Direct, take 2. Take 2. Types, ang tinatanong ko.
Pass muna, sir. Hinugot na sa stock knowledge.
Alright, sige, Alexis.
Hot and cold staining po.
Alright, very good. That's good. That's easy, right? Hot and cold methodologies are the types of staining methods that we have for AFB. Okay, so anong name nila? We have Kinyoun method, okay? And what else? Kinyoun method and Zil-Nielsen, okay? Kinyoun method and Zil-Nielsen, okay? Who's hot method and who's cold method? Zil-Nielsen or... Sorry, who's hot method? Zil-Nielsen or Kinyoun? Maria, Christina, Kai, Sabado. Okay.
Nielsen po, yung hot.
Pilsen? As in the brand of beer?
Nielsen po.
Okay, so I thought remember at the start of the lecture, I want you guys to be specific. Bakit? Bakit na naman? Kasi baka mamaya yan lang isulat ninyo, mamalian ni Sir Marco yan. Okay? Wag din ZN method ha. Alright, thank you so much. That is correct. Okay? Zil-Nielsen method and Kinyoun method. So ano naman pinagkaiba ni Zil-Nielsen method sa Kinyoun method? Usually kasi si Zil-Nielsen kailangan mo pa magpa-steam. Okay? Kailangan mo pa siyang steam. Si Kinyoun method, bayaan mo siya dyan matuyo. Ganun lang. Wash, and then bayaan mo na siya matuyo. Kaya cold method kasi room temperature lang siya. Actually kasi ang pinagkaiba lang naman nitong dalawa is this one uses methylene blue as the counter stain. And Kinyoun method, most of the reagents that they would give you if it's Kinyoun method is malachite green. Pero not necessarily that way all the time. Okay? Alright? So depending on the manufacturer pa rin siya. Pero just to give you guys a bit of a differential, just to give you a bit of history, you need to use a water bath or a steam bath, or if you don't have that, you can use your alcohol lamps. And when you see smoke emanating from the carbon fusion, that means that your primary staining is complete. Wash it out, do the colorization, and do the next step, which is your secondary stain. Usually it's methylene blue. Okay, all right, clear, cool beans. Let's move on to the next slide. Okay, gutom na ba? Hindi pa. Hindi pa. Okay. So, let's move on to the next question. What mordant or intensifying technique or intensifier technique is used in each of the two types of AFB staining? Oh, kakasabi ko lang kanina, 'di ba? Steam or heat for Zil-Nielsen and Tergitol for Kinyoun method. Okay? Cool? Understood? So kasi 'di ba, may usually Tergitol is incorporated in the primary stain. Okay? You would see methylene blue plus Tergitol. So okay, I know this is cold method. I don't need to heat it. Okay? So when you see reagents that says carbon fusion plus Tergitol, Kinyoun method ang gagawin ko. All right? So you don't need to heat it up. Baka sumabog. Charot. Charot lang. Walang ganon. Okay? Alright. Now, what decolorizer do we use? Or what decolorizer is used in AFB staining? Sige. I want specifics again, ha? Hindi pwedeng walang percentages. Marivic Sastre. Marivic.
3% acid alcohol.
Very good. Acid alcohol. Now, how do we prepare it assuming we need 1 liter? Ayan na. Sige. Ayan na. Remember, when I was an intern, we used to make acid alcohol. Would you believe it? Ganun na talaga kayo katanda, sir. Hoy! 32 and thriving pa ako. Sa December pa ako magiging 33. Okay? 32 and thriving pa ako. Wag nyo akong ginaganon. Okay? So, pwede pa din. How do we prepare it? Before we begin, what is our solution? What is our solute and solvent? Since you're talking about 3% hydrochloric acid, your solute is hydrochloric acid. Your solvent is what? The alcohol. Okay? Now, sir, lumalabas ba sa exams yan? Yes. Lumalabas yan sa exams. Okay? There was a student of mine, I think in 2015 or 2017. I think this was the second time I came back to the Philippines, and they gave me, and my students who passed the exam, they gave me some questions that was confusing for them. So I added this in this lecture for you guys, because they asked them, how much solvent do you need to make 3% alcohol for AFB staining? Assuming that your stock needs 1 liter. 3%. Ang solvent mo is alcohol. 'Di ba? Ang solvent mo is alcohol. Ngayon, ang solute mo 3% lang ang kailangan, hydrochloric acid. How much do you need to make a 3% alcohol solution? Gina, tutulungan ko na kayo. Tiret na. Magbibigay pa naman sana ako ng 500 GCash. O sige, si Diandra. Naku, ito talaga si Chosen.
Biglang nagtaas ng kamay. Iba talagang tawag ng pera.
Diandra, sige.
Ano po, 30 ml.
Sandali. Bakit hindi ka na naman nagpapakita? Ayan. Okay. Diandra, how do we prepare 1 liter of 3%?
30 ml HCl po.
Plus?
Tapos, 970 ml ethyl alcohol.
Very good! Oh my God, may nanalo, Sir Marco. Basta talaga pag pera, motivated talaga siya. Deandra, I need you to email your number to me, 164 at gmail.com. This is my email. Okay? I need you to email your number. I'll call my secretary later and I'll send you 500 Gcash. Okay? Okay ba? Alright?
Thank you, sir.
Okay, very good. Alright, so one of the three prizes, actually 1.5 ang ano ko sa inyo eh. 1.5 ang budget ko sa inyo. Nakasave na kaagad.
Pag naubos yan, like a gems na lang yan. Bahala
Oo,
kayo.
Bahala kayo dyan, like a gems. Pwede din naman kung papayag si Sir Marco plus, di ba? Di ba, no?
Ay, mahirap yan.
Mahirap.
Pag exceptional yung tanong and exceptional yung sagot.
Or exceptional activity like what Mark Segi is doing, right? Something that
Mahirap yan sa buhay. Pag ganyang moderator. Alam mo, madaming hihingin na tulong yan sa'yo si Manuel.
Oo. Magmo-moderate ka sa akin every time. Magsisend ka lagi ng links para sa kanila para dumami yung ano ko. Tapos isi-share mo para dumami yung mga subscribe. Charing, joke lang. Joke lang. Alright? Okay. So, that is correct. 30 ml. Sorry, this is small letter ml ha. Hindi megaliters yan ha. Milliliters lang yan. Okay? I think this is because of the slide the slide template. Okay? So 3% ethyl acid alcohol. This was a question in the board exams, by the way. If you need to write something that is important, if you want to write something that's important, please remember number 23, because this is a recall question. Okay? Now, what is the most commonly used counter stain in AFP? Okay, so I mentioned two, by the way, earlier. Methylene blue and malachite green. The most commonly used in many laboratories is methylene blue. Okay? Wala na. Hindi ka na pwedeng magsagot kasi hindi na ako nagtanong. Yung mga mahirap na questions sa mga tinatanong ko, mga computations, yung kahit ako hindi ko masasagot, pero nakalagay na doon yung formula para masagot. Okay. All right, let's move on. What other acid may be used as a decolorizer for AFP staining? Also indicate the percentage. Sige. Who here knows the answer? What other acids or acid can be used? Isa lang naman siya. Alright, I'll give you a clue, guys. There are only two types of acids that we have usually in the laboratory or in the clinical laboratory, and this is the other one that you have. Okay. Minty Fresh.
3% sulfuric acid.
3%? Are you sure? Are you sure about that? Girl, are you sure about that?
Yes po. Pag 1 liter, yun din po. 30 ml po din.
Dahil wala man ako tinanong ng preparation.
He's talking about the percent.
I'm talking about the percentage.
He's talking about the percentage, the concentration.
How much of the percentage do you need? I'm looking for a specific number. I'm not asking for the preparation. Sige, Marivic. Let's go, Marivic.
1%.
Nasaan ka muna? Bakit wala ka? Ayan. Mariva?
1%.
Alright, 1%. That's correct. Now, Marivic, since you raised your hand, what is the purpose of using this type of decolorizer? What's the purpose? We're staining for specific bacteria, right? What specific bacteria are we talking about?
To show OOCs appearing as well.
OOCs?
Wait.
What's OOCs?
Wait lang. Hindi.
They're studying verbatim. Ako. Parang feeling ko note si Dr. Llanera yan.
Rodriguez.
Rodriguez. Nako.
OOCs po is parasite ha.
Hi. Bacteriology tayo, guys.
Conidia.
We're staining conidia. We're staining conidia and microconidia. Nakakaloka.
Christopher Cryptosporidia. Charot.
Hi.
And
Okay, so if I may interrupt, no? So Rodriguez po is a decent reference,
For clinical
but
chemistry.
it's not been updated for a very long time.
Oo.
So there would be a few inconsistencies there.
Mm-mm.
So I'm not saying mali naman si Dean Rodriguez, no, but
We love her
medyo
to
luma
death.
na po kasi yung book na yan.
Mm-mm. We love
Okay,
her to
so
death.
if you're
Technical
going to use
team.
that as don't use Rodriguez as a reference, rather review book lang yan.
Mm-mm. Correct. But we're very thankful for Dean Rodriguez because she helped us out in many trials and tribulations when I was studying for clinical chemistry. But for microbiology, I'm sorry, ma'am. I'd go with Apollon. The exact questions. All right. So, what's the purpose? Yeah, she said she has an idea, but she could not it wasn't well executed. Who can help her out? It's used for sirit na, wala nang bayad, wala nang ano to. 100 peso Gcash sana. Wala, sirat na, dinugo na yung ilong ni Marivic eh. Her nose is bleeding. Wala na? Okay. We're done. We're going to move on with the function. So 1% sulfuric acid is actually the one that we need. She's correct. Now the purpose of that one is to stain or demonstrate weakly acid-fast bacilli. Weakly acid-fast. Hindi yung weakly as in linggo-linggo ha. Huwag kayong pilosopo. Weakly as in yung mahina. Okay? Alright? Hindi yung walang pilosopo dito ha. Baka mamaya sabi ni Sir weekly, so linggo-linggo ko daw gagawin. Hoy! Huwag ganon, okay? Weekly as in mahina, W-E-A-K. Alright? W-E-A-K. Tama ba spelling ko? Yeah, weekly. Okay? So ano ba yung mga weekly, natin? Mga linggo-linggo natin na organisms na kailangan ng 1% sulfuric acid. It's actually Nocardia asteroides. Okay? There are other species of Nocardia, but I just generalized it to Nocardia. Okay? All right. Now, the ideal smear for AFB is actually thumb size. In many of our old references, if I may say so myself, it's actually one of the things that Dean Rodriguez actually wrote in her books. It's actually thumb size, but yeah, she has a correct assumption of that one. But if we want to be specific, actually in the books, they are going to ask you the specifics of this smear. It's about 2 to 3 centimeters. 2 by 3 centimeters, rather. So it's almost thumbprint. It's almost like a thumbprint. Okay? Almost like a thumbprint. Okay? So be mindful of that one. Totoo ba to, sir? Kailangan kong tandaan to. Yeah. Because they're going to ask questions like that in the Philippine boards. For ASCP, not so much. More on your interpretation of the stuff that you've studied. Like for example, case presentations. Mostly those are the things that they're going to ask you for ASCP. For Philippine boards, since I was taking and since I was also the person that people come to when they pass, I'm pretty sure that the main differences is basically on memorization. Okay? So sometimes they would ask you, if there is a 2-angle tilt in your ESR tube, by how much will be the increase in this or that? Mga ganong keme. But that's for hematology. That's another headache for you guys. I would not dare intrude in that discussion. Okay? Now, what is the function of the polysaccharide component of bacterial cells? So I mentioned earlier that it inhibits phagocytosis. And organisms that contain this include 3. Okay? These include 3 organisms. Okay? Haemophilus influenzae, Neisseria meningitidis, and Streptococcus pneumoniae. What is the main commonality between these 3 organisms? Sige. Sino ang makakapagsagot? What is common with these 3 organisms aside from them having having a polysaccharide component in the bacterial cells? They are all what? They are all type ko ba? They are all causative agents of what? Of bacterial They are all causative agents of bacterial M. Bacterial Manuel de Leon? Sige, Nigel.
Bacterial meningitis po.
Very good. Very good. Very good. Very good. Bacterial meningitis. Tama ba spelling ko, moderator? Moderator, tama ba?
Yes po.
Tama naman, no? Meningitis. So,
Opo.
kita mo yung mga extra efforts na gagawin mo para sa akin. Di ba? Kailangan mo gawa ng spell check. Kailangan mo, pantayin yung video ko. Di ba? Ganon. Okay? So, that's the commonality between these three organisms. They often cause bacterial meningitis. Okay, so we're talking about bacterial meningitis, but what about another organism that causes this is not a bacteria, by the way, this is a fungus, okay, that has a polysaccharide component in the bacteria, in their cell wall or in the cellular cytology. I'm talking about a fungus that is capable of causing fungal meningitis. Sige nga, since we're tackling microbiology, and I'm also the one who's going to deliver the speech to you guys, the lecture to you guys. It causes fungal meningitis. Okay? Starts with letter. Who's there? Who raised their hand? Eliza. All right, Eliza.
Cryptococcus neoformans po.
Very good. Very good. I love it. Pero sayang, hindi ko pa nasabi na may prize. Sayang. Buti na lang hindi pa ako nakapag-promise, Sir Marco. Sayang. Sayang. Buti hindi pa ako nakapagsabi. But thank you so much. O sige, para malaman mo, para mabigyan kita ng 100, ano ang gram stain appearance niya? Ay, Cryptosporidium. Gram stain appearance. You will see it in page 295 of your analysis of urine and bodily fluids. 209. Oh, 'di ba sinabi ko na din yung page 229. 'Di ba? The topic of cerebrospinal fluids. 'Di ba? Ano 'yon? Ano ang gram stain appearance? Or gram stain morphology. Let's call it morphology if we want to sound fancy. So because we're talking about gram stain naman eh.
Gram positive po.
Yeah, but I'm talking about morphology, not the gram stain reaction. Yes, you are correct. It's gram positive. It's a yeast. It's mainly a yeast cell. But there's a specific terminology for the morphology. I'll give you an example of morphological structures, okay? What do you call this? Vibrio cholerae, coma bacillus. Diba? Yersinia pestis, safety pin bacillus. Right? Haemophilus influenzae, school of fish. Right? These are morphological structures. So I'm looking for the morphological structures or the morphological appearance. Right? Starts with letter S. Wala, siret na. 5, 4, 3, 2, 1. Nobody?
Siret.
Starburst. Okay? Starburst appearance. Okay? So what does starburst look like, sir? Yan ang bilog. This is what it looks like. Starburst. Parang nagsisabog lang. Okay. Yan. And what makes it different? Kasi merong line lagi sa gitna ng original. Okay? Parang ganun siya. Parang it's like a supernova. It's exploding. If that makes sense. Okay? It has a starburst appearance. Okay? Shall we move on? Alright? May natutunan? Okay? Are we ready to move on? Basics pa lang to, guys. It's basic review. We're talking about bacterial physiology as well as laboratory tests associated with it. All right. Now, but in Bacillus anthracis, what is the component of its cell wall? I think this is hard. It's a hard question for you guys. So I'm going to leave it. I'm going to answer it for you guys. It's actually a polypeptide or polymer of delta glutamic acid or glutamic acid. Sorry, glutamic acid. So polypeptide or a peptide of glutamic acid or delta glutamic acid. The polypeptide. So it is a component, a chemical component. Okay, it's the chemical component of a Bacillus anthracis cell wall. All right. Now, the site of production of energy in bacteria. Oh, sir, meron ba silang mitochondria? Wala. Right? They don't have mitochondria. So the site of energy production in eukaryotic cells. What cells do you have? Mark. What type of cells do you have? Prokaryotic or eukaryotic?
Sa be, being a human, sir?
Yeah, as a
Alam ko.
human being. Unless of course you're from
Sir, di ko,
from a
Di ko-
Suspicious yung tanong.
di ko sure, di ko po kasi malala, pero guess ko po eukaryotic.
Okay. Sir Marco, does he have to guess? Is it okay for him to guess whether or not he has eukaryotic
No.
cells?
Nakakagalit yung tanong niya.
Nakakagalit, di ba? Nakakagalit.
Nakakapanginig ng laman.
Nagagalit si
Sorry
Sir
po.
Marco sa mga ganyang bagay, Mark. Because it's basic biology. I think this is chapter 1 of What is Life. I think I have a book there. What is life? The difference between a microorganism and a macroorganism, di ba? So you have eukaryotic cells. You're correct. So the energy production of somebody who has eukaryotic cells or an organism that is eukaryotic is in the mitochondria. Now, what about for bacteria? Meron ba, sir? Yes, diniscuss yan. Hindi lang siguro kayo nakikinig. Ano, saan? Who can answer? Who? Who was that? Nigel.
Cytochromes po.
Cytochromes. Okay, yes. She's kind of correct, but it's actually in the cytoplasmic membrane. But I'll accept her answer. Okay, so sa cell wall mismo. Okay? Kasi yung nagre-react, nandoon mismo. Okay? Ganun. Kasi yung mitochondria mo, parang actually it's, if geneticists are going to be asked this question, bacteria is actually, the mitochondria is actually an evolutionary form of microorganisms. Kaya nga parang kidney, parang bean-shaped din siya lagi, di ba? So the exchange of ions actually occurs in the cell wall. And the exchange of ions actually produce a chemical reaction resulting from ATP, from ADP becoming ATP. So that's how they produce energy. Okay, so we're not gonna go and delve deep into that one because they rarely ask it unless you become a med student. So for the purpose of differentiating the two, I just added it here because why? Bacteria is a prokaryotic organism and Mark is a eukaryotic organism. Right? So alam mo na kung saan, Mark? Saan ka nagpo-produce saan nagpo-produce ang cells mo ng energy?
Yes po.
Saan?
Mitochondria po.
Very good. Okay. Hindi na galit si Sir Marco. Galit ka pa, Sir Marco? Nanginginig pa rin laman mo?
Slight lang.
Slight na lang. Humanda ka sa exam, Mark. Pasalamat ka, may plus 10 ka daw. Oh, di ba? Alright, now, this is the point of which the genetic material of bacteria is attached. So again, this is something that you need to talk about. Bacterial cytology is at the topic of bacterial cytology or bacterial physiology. Okay, so we're just going to lay the groundwork for you. Okay, so in microbiology for medtechs, we don't really discuss the specifics of the physiological nuances of what happens in a bacterium. What I'm more focused on for you guys is the structures. So it's on the mesosome. Okay? It's basically like an attachment. If the best example that I can give you guys is like, hang on a sec. Where is Paint again? Can you guys see Paint? Favorite kong application ngayon is
Yes, sir.
Paint. Okay, so for example, this is a rod-shaped organism. I know it doesn't look like a rod. Okay, so the insides of that one, this is where merosomes are located. The merosomes is basically like a point in the bacterial cell wall, and usually this is where it happens. Why on the side, sir? And usually you wouldn't be able to stain it, but microbiologists have seen and noted this using electron microscopy, okay, that they've seen certain structures at the edge of cells, and this is actually the site at which the other cell for binary fission actually goes out of. Kaya doon yung malalaman yung mga merosomes natin. Okay? So merosomes, okay? Clear? Clear ba? Minty Fresh, clear ba?
Yes po.
Huwag ka pa smile sa akin, Minty Fresh ha. Alam ko inaantok ka na. Smile ka lang sa akin. Tigilan mo ako ha. Ikaw din Nigel, pangisi-gisi ka lang ha. si Alexis lang yung seryoso ang datingan, oh. Hindi siya matitinag. Hindi siya matitinag sa pagkaseryoso niya talaga. Natutuwa ako ngayon kasi nag-video call sila lahat. I love it because it makes me feel like I'm actually talking to people. Unlike the last lecture that I had, we rarely had some interactions because I couldn't see their faces. Okay, now let's talk about inclusion bodies. So medically important inclusion bodies for medically important bacteria are as follows: Much granules. Okay, we have Much granules. We have Babes Ernst granules and bipolar bodies. So where are they found usually? So Much granules, you already heard it before. Probably you might be familiar that it's found in Mycobacterium tuberculosis. Okay, Babes Ernst granules usually is known as metachromatic granules or volutin granules, usually seen in organisms like Corynebacterium diphtheri. Okay, and the other one is Yersinia pestis. Okay, Yersinia, hindi Yersina ha, Yersinia. Okay, all right. So bipolar bodies, making it look like a safety pin bacillus. Okay, so I always mention it that way. All right. So what stain is used to demonstrate bipolar bodies? Oh, sir, do we need to know that? Yes. Unfortunately, you have to remember this. Wesen stain is a way that we can use to demonstrate the bipolar bodies of Yersinia pestis, making it look like a safety pin bacillus. Ano bang itsura ng safety pin? What do you guys think of when you're looking at a safety pin? What do you guys think of when you look for a safety pin? Di ba ganito din yun, yung safety pin? Wait, ibahin natin yung color. Right? It's something like this. Humihinga ng malalim. May humihinga ng malalim sa background. Medyo napapagod na. So it looks like a safety pin kasi 'di ba? Ganyan yung itsura ng safety pin, 'di ba? Sasarado mo lang dito. Yung bukas sara.
Yes po.
'Di ba? Pardible. 'Di ba? Tagalog pardible. Like, I remember when I was a student, one of my colleagues or one of my classmates, she's wearing a very ill-fitting, I would say tightly ill-fitting uniform, similar to what Alexis is wearing. Are you wearing a scrub suit or something? Is that a scrub suit or something? Imagine it 10 times tighter, and she went down and her pen fell on the floor, and she took it as if she was wearing something that is actually her fit. What happened was that it exploded. All the buttons exploded. And she used pardible. Diba? Yun yung safety pins. Okay? Baka kasi sa sobrang ninyong pag-aaral, nakakalimutan nyo na kung anong itsura ng safety pin. Alright? Next. Kasi alam ko nag-study kayo lahat. Napaka-active nila eh. Yung class ni Diandra, onti lang ang nagsasalita doon. Diyos ko. Kung hindi ka pa kung hindi ka pa magwagayway ng pera na may GCash, ano yon, di ba, Marco?
Sir, pagod po sa internship.
Ha? Ano?
Pagod po sa intern.
Tigilan niyo ako. Ganyan talaga kayo. Buti pa to, meron tayong Minty Fresh, meron tayong Nigel, meron tayong Alexis, 'di ba? Meron tayong Maria Cristina, kaya ang haba talaga ng pangalan mo, girl. Nahihirapan talaga ako sa iyo. Pwede bang McKay na lang para social? Can I call you McKay? Right?
Any will do, sir. Pero
God, I
mas
love it.
preferred po nila Sabado.
Sabado? Ayoko! Kasi ibig sabihin nun may pasok na ako pag Sabado. Girl, wag. And it sounds so tita. Really, it sounds so tita. As in, Sabado. Diba? I'd call you McKay. It's very California girl. I love it. Right? Gusto mo yarn? Diba? Gusto ko yarn. Diba? Gusto ko talaga tong class na to kasi nagpapakita sila sa ano. You guys are now my favorite. Yung class ninyo nila Deandra, sila Adrian, nako, kailangan pang wagaywayan ng pera para magsalita. At least kayo, nagsasalita kayo. Ganun. Diba? Alright. Now, what do we use to stain the volutin granules? We use the Lamb's method or Burke's method. And the main thing that you need to remember when you're staining volutin granules is the last question in this slide. What stain is used? It's actually methylene blue. Now, Lamb's method is different because you need to use heat. For Burke's method, you just need to let it dry and wash it off. Okay? So that's the only difference between the two. It's actually like Zell-Nielsen and Kenyuan. Actually, you don't need to remember. But what you need to remember is that when you want to demonstrate, usually questions will appear like, which of the following coloring agents is used to demonstrate volutin granules from Corynebacterium diphtheriae? You would just say methylene blue. If I see somebody answer carbol-fuchsin or safranin, nako, kukulamin ko talaga, ha? Wag ganon, ha? Mambabarang ako. Wag ganon. Kaya mambabarang ako ng tao talaga kapag kami sumagot ng safranin. Okay? Alright. Next. In number 34, this is a structure found in some bacteria that is considered as a resting cell. This allows the organism to survive in extreme conditions. Who can answer that question? This is a structure in some bacterial cells or in some bacteria that is considered as a resting cell. This allows the organism which allows the organism to survive extreme conditions. From the word extreme condition. Sige, Alexis, talagang seryoso si Alexis pag-aaral niya. Gusto niya one take lang talaga siya ng exam ni Sir Marco. At saka gusto niya sa kanya lumilibot ang fate ng mga klase niyang babagsak. Sige, kasi pag highest, sa kanya magiging 90, lahat hahatakin pataas. Hindi pwede pakakuha ng 10 pag nakuha ni Alexis ng 100 kasi automatic bagsak ka, walang considerations. Ganun. Sige, Alexis.
And spores po.
Very good. Very good. Spores. Now, what is the chemical component of the spores? Kailangan talaga may chemical component, sir. Bakit kailangan pahirapan kami? No, you need calcium dipicolonic acid or dipicolinate. Okay? Or dipicolinic acid. Okay? Dipicolinic acid. Endospore or a spore, I would accept spore. Okay? But if this is mycology, it's important that you answer endospore. Okay, if we had a mixed discussion of microbiology and bacteriology, I need you to answer endospores. Okay, because there's a lot of spores for fungus or a fungus. Okay, now examples of medically important bacteria that are considered as spore formers. There are only two. Okay, so who here is knowledgeable enough or have enough stock knowledge? Valiant. Okay, hindi talaga nagpapatalo ang mga kabaro ko. Sige, go!
Bacillus and Clostridium.
Taray talaga. O sige, hindi kita titigilan. Kasi ayoko nang napapahiya tayo. Wag mo akong ipapahiya, ha? Nakadepende ang kalidad natin as mga students. Valiant, ha? Minty fresh. Okay? So, what is the organism or spore locations for each organism? So, sabi mo Bacillus tsaka Clostridium. Sige. Let's start first with Bacillus. Where is the location of the spores for Bacillus? Gigigil ako sa'yo. Charot lang. Ano ba? Kakainis ka. Sayang pa naman. I thought we are on a roll. Talagang nasa iyo pa naman ako. Sa iyo ako nakadepende. Kasi sabi ko, there is something with this kid. He knows his microbiology. Who raised their finger? Her finger? Her hand pala. Her finger. Alright, who raised their hand? Somebody raised her hand. And it was a girl's name. I wasn't able to catch it. Who was that? Who said that? Sino yun? Hindi ko nakita eh. Sorry.
Sir, pwede i-try?
Okay, sure.
Ako po yun.
Okay, Marivic.
Yung ano po, yung Bacillus, nasa central po. Central
Okay.
spore.
Very good.
That's
Very good. Proceed.
Clostridium. It's either sa terminal or subterminal.
Okay. Now, balikan natin si ayoko talagang natatalo, mga Becky. Hindi ako papayag. Thank you, Marivic. More power to the women. Pero babalikan natin yung Minty Fresh. Where is the location for Clostridium tetani? And where is the location of the spore? Where is the location of the spore for Clostridium tetani? Where is the location of the spore for Clostridium botulinum? Clostridium, other Clostridium species.
Hindi ko
Sabi
na.
ni terminal. Nako, natatakot talaga ako. Oh my God.
Ang ganda na po sa knowledge.
Ikaw ang future natin. Bakit? Sa'yo ko pa naman ipapasa ang baton. Nakakainis ka. May makeup pa na lang kita, girl, pag umuwi ako ng Pilipinas. May pasalubong ka sa akin, ha? Okay?
Sige
Alright.
po.
Okay. So, para sa kaalaman mo, ha? Talagang sa kanya ko lang i-explain. Para sa kaalaman mo, for Clostridium species, for every Clostridium species, it's subterminal. Okay? Sorry. It's it's subterminal. For Clostridium tetani only, it is terminal. Okay, so I actually have a drawing for that one. Okay? So everybody else has a terminal spore except for Clostridium everybody else has a subterminal spore except for Clostridium tetani. Kasi siya yung nasa dulo. Okay? Pag sinabing subterminal, somewhere here. Okay? Somewhere here, but not at the very edge. Okay? Making it look like a drumstick bacillus. Okay? Okay? Gets mo na, Minty? Okay? Bigyan na lang kita ng makeup, ha? From Trixie Mattel. And Patrick Star. Okay? Alright. Okay. Huwag ka mag-alala. You're still one of my favorites kasi isa ka lang na Becky. Unless lang mag-Becky si Mark, which I don't think is highly unlikely in the rest of at the end of our lecture. I don't think so. Unless of course may tinatago kang sikreto, hindi namin alam, whatever. You do you. We're here to support you, right? Right, Minty? We're here to support you. Anyway, let's go. Okay, sabi ko sa iyo, Mark, magtitripan kita later. Examples of stains used to demonstrate endospores. Ayan na. Okay? So, madami tayong names na pinapangalanan ngayon, no, sir? Diba? Nakakainis. Pero, kailangan ba naming aralin, sir? Yes, of course. Because there will be a question. Maybe I'll be the one to prepare it. Maybe it's Sir Marco. I don't know. But there will be a question there that will ask you, which of the following is a stain that demonstrates endospores? So you gotta be able to answer either of the following: Schaefer-Fulton, Ritz-Conkin, or Dorner stain. And then I'll add somewhere there. I'll modify the question, or maybe Sir Marco will modify the question. Which of the all of the following are stains used for demonstrating spores. Then nilagay ko dito sa baba, India ink. Tama ba yon? Di ba mali? Napakadali, di ba, pagka ganun, Sir Marco? Gusto ko yung nakakalito, yung Wayson stain. Lalagay ko dito Wayson stain para makalimutan talaga ng mga students. Di ba? Dapat pangalan din ng tao. Kasi madali lang pag
O kaya
nilagay ko
none of the
India
above.
ink. Di ba? O kaya lalagay ko none of the above, all of the above. 'Di ba? Para lang sa ayoko naman medtech, gusto ko na lang mag drag queen. Ganun. Gusto ko na lang mag drag queen. Nakakainis. Ayoko na mag medtech, gusto ko na lang maging barista ng Starbucks. Nakaiinis ako sa iyo, sir. Ang dami kong kailangang alalahanin. Pero ganun talaga. Kung kami ni Sir Marco nakasurvive, kayo din kaya nyo. Okay? Mas mataas lang ang grade namin sa inyo. Ganon. Mas mataas lang. Di ba? Magandang kasabihan yun, Sir Marco, no? Di ba? Kung kaya namin ni Sir Marco, kaya nyo
Gagayahin ko na
rin.
yan.
Pero, mas mataas ang grade namin sa inyo. Ganon. Ganon dapat. Kasi walang mas tataas sa grade namin kasi kami nagtuturo sa inyo. Kapal naman ang mukha nyo kung mas mataas grade nyo sa amin. Manggigigil ako, lalo na kung si Minty Fresh yung may hanggang dun lang ang kaya niya sa may sa species. Di ba? Okay? Alright, next, let's move on. If you're going to use endospores to stain each method, if you're going to use stains to stain the spores, or endospores to be specific, there's color reactions. Actually, I don't really care much for this one, but it's added in my list of things for you guys. I wouldn't focus this much. I would focus probably on the names of the stain. Okay? Ritz Conkin and Schaefer Fulton will have green. Donor stain will be red. I think the easiest way for me to remember this is that Dorner has R and E and D, so red, while green is while the letter G is not found in Ritz Conkin and Schaefer Fulton. I don't know. You decide your own. I would call your mnemonic preparation as choose your own adventure, probably. Okay, now. Let's start with the structures. Structures that may be found in the external cell wall of the bacteria. This means that it's pointing outwards, okay? So it's pointing outwards of the bacteria, of the bacterial cell wall. It could either be a capsule, okay, or it could either be a slime layer. Slime layer is usually found in organisms that have a slime layer or a slimy layer. We'll talk about those organisms later on, but for now, what we're going to talk about is capsules, because capsules are responsible for the inhibition of phagocytosis. Again, I mentioned it several times now, right? Inhibition of phagocytosis. Now, I need to know 3 general methods in demonstrating bacterial capsules. There are 3. You could either you could either grow the organisms in milk media, you could stain it, or you could use serotyping using antibodies. Okay? All right? Is that clear? Okay? So, bacterial capsules. So you could grow the organism in a milk media, or if you want to grow it, you can stain it, or you could use antigenic serotyping. So this could be a question again. All of the following methods are used to demonstrate bacterial capsules. I said all of this. Then nilagay ko sa ibaba. Then I put at the bottom, animal inoculation using rabbit testes. Tama ba yorn? Diba? Di tama yon, di ba, Mark? Bakit nawala yung mga students? Sir Marco, bakit nawala sila? Na-disconnect ba ako?
Ay, hindi po.
Hindi sir, lutang na yan kasi alas 3 na.
Oo. Alas 3 na. Snack time na ba? Wait lang, malapit na ata tayo matapos with our first topic of the day. Lilipat din tayo. Huwag kang mag-alala. Huwag kayong mag-alala. There are still 300 slides to go, and we're going to try to finish it within the day. At least. Okay? Now, bacterial capsules, medyo nangongo. Bacterial capsules can be stained using HISS method, or the HIS staining, or nigrosin or India ink staining. Nigrosin or India ink is known as a background or a relief stain. So what are we staining when it's a background stain? You are staining the background rather than the organism. Okay? So his staining method. This one is actually a very complicated test. I looked at it when I was a student. I did not care for it because there's not a lot of questions about it. But nigrosin in India ink, there's a lot of questions about it. It's very high yield. Okay? Specifically, what organisms will test, which organisms will it test, what specific samples may require the need for India ink staining, something like these. Okay? Each organism has its own specific what do you call this? Has its own specific testing procedures to demonstrate, and in microbiology it's quite tedious, and that's one of the reasons why you would have to take 5 units of it, and even if you took the 5 units of it, you may not be able to understand everything that is fine. We're here to help you out. Okay, now let's move on to the agents used for his staining. So it's very easy. I made it easier for you guys to remember it. We're going to start with gentian violet, and then using basic fuchsin, and then a copper sulfate wash. But everything in between, there is some sort of step in between. But the main reason why I made it look like this is because you guys need to remember it similar to gram staining. But there are some additional steps in between. But if they ask you questions like gentian violet is used in which of the following staining methodologies for bacterial morphology, that's the time. Oh, okay. I remember. This is for his. So I need to use it for capsules. Alright? Ganda ba? Madali, di ba? Madali, no, guys? Huwag kayo mag-alala. Pwede kayo magkape katulad ni Mark. Huwag kayo mag-alala. Walang problema sa akin. You can eat in front of me as long as you're sharing it to me virtually. Okay? I don't really care. I don't really care that much. Actually, pwede ka kayo manood ng Netflix pero siguraduhin nyo lang napapasa kayo sa exam ko kasi I will only think of 3 questions every week starting this week, and I'll make sure it would be the most difficult exam that you will ever take. Okay? Okay, mag-alala. 3 question lang, tapos 1 linggong pag-iisipan ni Sir bawat isa. Oh my God, bakit ganun? Sabi kasi ni Sir Marco, maski siya daw mahihirapan. Yun yung request niya sa akin. Tama ba, Sir? Let me just confirm.
Yeah. Kasi, yung exam nila, yung diagnostic exam nila, it kind of looked like they weren't taking it seriously. So, I'm going to assume that they're very confident na alam na nila yung ginagawa nila. So, we'll give it the most difficult exam that we can come up with.
Yeah. Very good. 50
So,
items lang ba yun?
mukhang challenge nila ako. So,
300.
challenge accepted.
Pwede naman sa akin, Sir, 300. Kung gusto mo. Kaya ko yon. Siguro
300 items?
50 items every 2 weeks. Pag-iisipan ko yung 50 items. Diba? Ganon. And it would be something that you will not be able to see in any books. Diba?
Oo, hindi naman makakareklamo tong mga to eh. So,
bala kayo. Kasi hindi nyo daw sinunod.
Kasi
mukhang sa Sir Marco.
They look, diba? They look very confident now.
Alam mo naman ako, resback ako ni Sir Marco, diba? Ganon. Diba ba't parang naging tomboy yung boses ko? Sorry Sorry. Alam mo naman ako, resback ako si Sir Marco. Niloloko nyo daw eh. Hindi-etchos nyo si Sir Marco eh. Manda kayo, lalo si Minty Fresh.
Sal yan, ba't mukhang nagising ka? Nagising sa 300 items?
Actually, mas maganda nga yung practice na ganun, sir, di ba? Yung 300 items na sila. Kasi at least...
Yeah, that's true. For board exam, makasiguro 1,000 questions kayo per subject. Pasado na kayo.
Di ba? Diba?
Hindi pa rin sila nakakonvince yun.
Yung itsura ni Glenn, I'm not
having it. Ini-scam tayo ng dalawang to.
This is something that I did not sign up for. I thought I was studying to become a medical technologist. Sabi niya talaga, she is not having it. Itsura talaga ni girl! Ganun yung itsura ni Glenn. Nakita ko yung itsura talaga niya. Umiikot-ikot yung mata. Alright. So
kasi isipin mo, sir, 250 items, tapos nila 30 minutes.
Ay! Hala.
Galing, di ba? Ako, di ko kaya yun.
Ay, hindi nga nila sineryoso. So, sige, dadagdagan. I'm going to up the ante. Sige. Umanda kayo sa akin. Alam niyo naman ako, resback ako, Sir Marco. Hindi bagay, no? Last ko na talaga maging lalaki. Hindi ko kaya. Okay, let's continue na. Sumama ang loob ni Sir Marco sa inyo, lalo na yung mga sagot ni ano kanina na hanggang dun lang. Yung mga eukaryotic. Nako, humanda ka talaga ha. Humanda ka talaga, Mark. Okay, let's start with antigenic serotyping. Okay, so antigenic serotyping is used also to demonstrate a capsule, as I mentioned before. The positive result is basically ballooning, or what you call swelling test. And one of the tests that we use it for is known as what we call the Keelang test, or the Neufeld-Keelang test. Keelang test or Keelang test. Positive result is capsular swelling. So what happens? Why do we see capsular swelling? Well, basically it's an antigen test. What happens is that the antibodies that you add into the suspension will cause the cells to balloon up, basically by activating either the complement pathway, making it explode in the process if it's actually the organism that you're targeting. Okay, so we want to demonstrate the capsules. That's the basic principle of this one. And one way for us to do that is to add antibodies against a specific capsule. So it depends on the type of capsule actually that you want to demonstrate. But in essence, the name of the test is Keelang test, and that this is the positive result. Okay, clear? Clear. Keelang test. You will see it in our discussions in Streptococcus pneumoniae. Maybe in our discussions also probably about when we go to mycology, and if you discuss also in the future immunology and serology, if you pass this exam, or if you pass your exams, diba? I think it's for seminar 2, sir, right? If I'm not mistaken, you're going to take seminar 2 immunology. Anyway, let's move on to slime layer. The slime layer is observed in what species of Staphylococcus? Right? Slime layer. Staphylococcus what? There are 3 species of Staphylococcus. Who can guess for me? I know people are going to guess, so who can guess for me? Medical importance, of course. Staphylococcus of medical importance. Slime layer. There's 3. Oh, bibigyan ko na kayo ng choices. I'll give you guys choices. Okay? First one is Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. Which of those 3 has a slime layer? Alexis was the first one. Terribly sorry, minty fresh. All right, Alexis.
Epidermidis.
Very good. Yay! Finally.
Wow!
Yay!
Impressive na alam yun.
Very good. And the correct answer is Staphylococcus epidermidis. But she just said epidermidis. I don't like it when students are not completing their answers, right? Remember, you have to be very specific, not just in your work, not just in your academic work, but also in your work in the laboratory. Hindi pwedeng shortcut, okay? Ano nga sabi ni Sir Marco kanina sa akin, nagpapadeport ako ng mga medtech na hindi maayos ang grades, incident report, mga performance. We do performance checking, we do competency testing. Pag hindi ka talaga fit sa section na yon, you will be transferred somewhere else, and if you had 3 strikes with me, you will be transferred to a different department altogether, or a different hospital under the Ministry of Defense. Di ba? Ganon. Ang sama naman ni sir. Ganon ka talaga, sir? Hindi. Hindi ako ganun sa Pinoy. Siyempre, love ko yung Pinoy, yung mga ibang lahi. Charot! Ang sama ako! Charot! Nako, YouTube, baka alsin ako as hate crime. Ma-flag as hate crime yung ano ko. May hate speech. Let's remove that from YouTube. I'll edit it out later. Anyway, capsules in some bacteria are also synonymous to what antigens? We call it the K antigen or the VI antigen if we're speaking about it in what organism. Sige nga, VI antigen for what specific organism? VI antigen. Diba? Pumunot ang noo ni Alexis. Alam niya kaso hindi niya maalala. Nigel.
Salmonella, Shigella, and E. coli.
Salmonella lang, te. Excited ka, te. VI ang tinatanong. Advance talaga siya mag-isip. Pero I love it! I love it! Advance mag-isip. Pero sige, Salmonella what? Salmonella what? There's a lot. Salmonella typhus, Salmonella typhimurium, Salmonella cholera suis. Ano ba? Madami tayong ganon. Ano doon? O, sige, Alexis. Inakawan ka for the steal. Alexis, sige.
Salmonella typhi.
Alright, thank you so much. Alright, o ayan na. Ibibigay ko na para kay Nigel yung advanced pag-isip. I love it. Di ba? Kay antigen, capsular antigen for E. coli, Klebsiella, di ba? Ay, saka Klebsiella pala, sorry. Klebsiella, ganun. Okay? Okay? Cool? Cool beans? We can move on? Okay ba? Walang tumatango. Ayan, okay, sige. Tumango na sige. Yung moderator ko, hindi ko nakikita yung mukha, nakakabwisit.
Ay, sorry po.
Nabibis ko na si Adrian talaga. Pwede bang i-fire natin yung moderator? Joke lang. Char lang, ano ka ba? Kung mag-alala, hindi naman ganun kadami ang paggagawa ko sa'yo. Okay? Next. Nasaan na ba yung PowerPoint presentation ko? Next. Based on function, what are the two types of pili structures in bacteria? So basically, you have two types. Ordinary pili, which is just used for adherence functions, and we have what you call the sex pili. Okay? For bacterial conjugation. Totoo, sir. They do bacterial conjugation or they do segs. Sorry, baka ano tayo sa YouTube, ma-demonetize tayo sa YouTube. They do segs. Yes, specifically Neisseria. Okay? Yeah. One organism, one bacterial particle, or what do you call it, bacterial? Let's just call it a cell. Okay, since we're talking about cells anyway, one Neisseria cell will go to another Neisseria cell and basically penetrate it using its own sex pili. And the sex pili will serve as a function where the endosomes enter the other organism cell. And then they would divide and make a happy family ever after. Okay? So that's basically how they do it. Okay? How they do it talaga. I know it sounds a little too salacious, but it is how bacteria will do it. Bongga si Nigel nagbasa ng dyaryo. I love it. I love it so much. Mataas na ang palitan ng dull hairs. Kailangan ninyong mag-aral. Mataas ang palitan ng dull hairs. Who here are not going to continue? Who among you will not continue as medtechs? Will continue as doctors or physicians in the future. You're just taking up medtech, medical laboratory science as a pre-med. Well, you'd be wrong if you're going to take it as pre-med. Charot. Because right now, a lot of medtechs are migrating to the United States. They're offering a lot of money. There's even a 10,000 sign-in bonus for many companies in the United States. So this is high time for a lot of us to study. Okay? You just need to put yourselves into it. Okay? Okay? Okay ba? So next time, baka mamaya kayo makikita ko sa IELTS review naman. Iko-correct ko ang grammar ninyo. So talagang hindi nyo ako matatakasan. May IELTS review pa ako. And a lot of my students actually from CMCC actually enter my IELTS coaching sessions and they would say, "Sir, ikaw ba yung nag-lecture sa amin dati ng clinical microscopy?" Yes, that's me. Kasi ipapakita nila sa akin, sabi ko, familiar sa akin to, friend ko sa Facebook. Oh, di ba? Hindi nyo ako matatakasan hanggang career nyo outside of the US, outside of the Philippines. Ako ang dadaanan ninyo. Di ba, Sir Marco? Ako ang magiging bangungot ninyo.
Madaming racket niya si Sir Manuel Calanya.
Magiging bangungot ninyo ako.
Gatekeeper yan.
Oo, pagka nakita kong incompetent, ako lalo na kapag kukuha kayo ng what they call this? Kukuha kayo ng competency sa kapag nagtrabaho kayo sa Middle Eastern hospitals, talagang ako ang magiging ninyo. Ako talaga magiging bangungot ninyo. Kapag dito pa lang nakita ko hindi maganda ang performance ninyo, tapos hindi niyo siniseryoso si Sir Marco, I will be your greatest nightmare. Charot! Alright, now, stains used for demonstrating bacterial flagella. Okay, so what are the stains used? We have Leifson stain, gray stain, and Escher-Kohn. Ang dami naman, sir, but all of them have something in common. Huwag kayong mag-alala. All of them have something in common, and I put it in bold. It's tarmic acid. Okay? So you might be asked, which of the following chemical or which of the following is a component of bacterial flagellar stains? Dipicolonic acid, calcium permanganate, tarmic acid, or 1% hydrochloric acid. Obviously, what you're going to select is tarmic acid. Okay? Okay ba? 'Di ba? Ganun yung mga kadali yung mga question na mga makikita nyo sa ibang libro. Pero for me, hindi ganun. Alright? You are assigned as a research assistant in this and that hospital. Your boss or your laboratory supervisor asks you to prepare bacterial flagellar stain. Which of the following is the component of a stain that you want to prepare? Oh, ganon! Ganon yung mga tanungan. Di ba? O, di ba? Di ba? Di ba? Di ba? O kaya, the efficacy of which reagent is important in demonstrating bacterial flagellar cells? Di ba? Di ba? Ganon ang mga tanungan ko. Hindi ko kayo tatanungin what reagent is used to determine is common in bacterial flagellar cells. No, I don't think it's very common. Right? It's a very common question. It's very easy. Okay? And I don't like you guys to make it too easy to. Kasi hindi nyo daw sineryoso si Sir Marco. Alam niyo naman ako ang backup ni Sir Marco eh. Ano ako ni Sir Marco? Widis ako pagka may bumubwisit sa kanya. 'Di ba? 'Di ba? Yung binubwisit. Okay? Next, what are the type of bacterial flagella? Okay, so we have 6 types. So for 200 Gcash points, 6 types of bacterial flagella. I need someone to what do you call this? To volunteer. 6 types. I'll use a timer para at least fair tayo. Sige. Who will volunteer? Nag-unmute na si Mark. Sige, nag-unmute na si Mark. Sige. Gusto ko yan. Yan. Gusto ko yan. So, para maniwala si Sir Marco na hindi ka talaga, na sineryoso mo talaga yung exam kahit 20 minutes mo lang siya tinake. Sige.
Hindi po ako yun, sir, pero kilala ko.
The shade.
Atricus, Monotricus, Ampitricus, Cephalotricus, Peritricus, sa Lophotricus.
You made a mistake with Cephalotricus. What the heck is Cephalotricus? What's Cephalotricus? You made a mistake. Alright, somebody else help out our friend Mark Segi. James Paul.
Atricus po.
Okay, 1.
Monotricus. Lophotricus. Ampitricus. Peritricus. Amphilopotricus.
Okay. What else? There's one more.
6 po. 6 na po yun sila.
Kasi may mali dun sa isa.
Bakit may dagdag bawas yung mga sagot nila?
Kasi may mali dun sa isa, kaya naghahanap ako ng isa pa. Diba? It's a choose your own adventure kind of a question. Sige. Try to correct yourself with what the one thing is you've said one thing that was mistaken. Sige, bigyan kita ng chance. Baka sabihin mo, tapos sabihin na naman ng mga babae, bakit pag lalaki, sir, may chance? But pag kami wala, eliminated agad. O, sige, bilisan mo na. Sige.
Opo, ulitin ko po.
O, sige, ulitin natin. Sige.
Atricus po.
Okay.
Monotricus. Lophotricus.
Okay.
Ampitricus.
Okay.
Peritricus.
Okay.
Tapos, Amphilopotricus po.
Oh, I'm terribly sorry. Talagang hindi talaga natin nakuha yung tamang sagot. Eliminated. I'm so sorry. Okay. Glenn. Ayan na si Glenn. She is not having it. She knows that the answers are incorrect and she is here to save us all. Alright. Go.
Atricus, Monotricus, Ampitricus, Cephalotricus, Lophotricus, and Peritrocus bacteria.
Huh? Bakit? What is cephalotricus? What is cephalotricus? Ano yung may ulo? May cephalid.
Yun nga, bakit may dagdag bawas yun sa kanila?
Oo, what is cephalotricus? I'm curious. Can you please describe what is cephalotricus flagellar structure? What is a cephalotricus flagellar structure? Oh, hindi. Hindi niya ma-describe.
Um, um, this bacteria have a tuft of flagella at one end.
That's Lopotricus.
Really?
Tama.
That's,
Sorry.
that's
Ayun po
Lop-
kasi yung nakalagay dito sa notes ko. So, mali.
Kanino na naman yan? Kanino
Ay,
man na?
ano lang sa past school ko. Sorry.
Okay. Alright. Okay. Who else? Valiant daw. Sige. Minty Fresh. I love it. Gumanon pa talaga. Isa pa. Project. Project.
Sir,
Walang crash
I
prize
am
pag hindi
serving
nag-project.
you.
Walang ano.
Sir, I am serving you Atricus, Monotricus, Ampriticus, Lophotricus, Bilofotricus, and Peritricus.
Anong kagulo? Sir Marco, anong kaguluhan to? Sir Marco, saan nila nakukuha yung Amphipetritricus?
I don't know. Actually, I don't know what they're saying.
Yeah. O sige na.
May
Wala
dagdag bawas
nakakuha
lagi
ng
na isa.
prize. Wala nakakuha ng prize. Everybody else is correct, but this is the answer. So, Atricus, there's no flagella. Okay? Monotricus, one pole of flagella. Amphitricus, poles on both, two on both sides of the on both, basically. Lopotricus is basically a tuft of flagella. Okay? Peritricus is all over the organism, hence the term peri. And periplasmic flagella or axial filaments, usually seen in spirochetes. So, yorn. Okay? So, hindi ko alam kung ano yung cephalopotricus. Saan yun?
Sir, yung
Sorry
ano
naman.
niya, yung
Oo, kasi si
structure
Mark
niya
din
parang
Huwag ka mag-alala. Si Mark din kasi
may-
may ganun
may ano po, tuft of flagella in both pole sides of the ano.
Okay, can you give me an organism with a cephalopotricus type of... Could you give me? Because in these things, they all have examples.
Okay.
Siguraduhin mo medically important yan. Baka mamaya
Kaya yun lang
yan
po yung nakita ko. Pero
Google.
Opo.
Okay.
May nakalagay po, Pseudomonas.
Pseudomonas? Lopotricus si
Sa internet.
Pseudomonas. Ay, Amphitricus si Pseudomonas. Totoo ba? Parang iba. Okay.
Class, kapag nag-google, no, wag niyo masyadong pahalata that you're just actually googling that at that specific time. So, medyo tingnan niyo muna kung tama yung ginugoogle niyo.
Pero in fairness, at least may sagot si Glenn at saka si Mark na halos parehas. Just so happens na wala siguro silang maibigay na organism. Kung pwede nyong i-defend sa akin, give me an organism, okay, sure. Give it to me. But as what another one of your classmates says, Pseudomonas is cephalopotricus? I don't think so. It's Amphitricus. Okay? In many of the books that I've researched on. Okay? So, enough about that. They would ask you questions about the locations of the flagella of the organisms. So, these are part of the basics. Okay? So, please be mindful of these things. Okay? Now, let's look at the function of flagella. Basically, the function of flagella is for motility. Now, this function is best observed at what temperature? Yeah. Is it freezing temperature, body temperature, or at room temperature? Sige. I've given you guys choices. I need participation. Let's try and use the chat box, guys. So, para mabilis ang pacing ng ng discussion. So, your choices will be for the second one is room temp. You could just write RT. You could say 37 degrees for body temp. You could say also you could just say freezing temperature less than 0, or you could say 0 degrees. Okay, write it on the chat box if you want, and we'll check it out later. Okay? Medyo na-realize ko malapit na pala matapos yung discussion natin. Hindi pa tayo nakakalahati. Let's talk about motility is best at 25 degrees Celsius or at room temperature. Okay? I should have skipped that, but whatever. At 37 degrees Celsius, the motility of some organisms are inhibited. What are the examples of these organisms? So we have two. Okay? We have two organisms. And I'm going to skip number 50, and I want you guys to answer this question because it's very important. Okay? It's important because when we talk about pasteurization, these are the organisms that you need to remove because of their motility. Okay? Because of their motility. Okay? You need to kill these organisms in pasteurization because when they are motile, they are highly pathogenic. Therefore, inhibiting them by increasing the temperature for quite some time, as in the case of pasteurization, is important. So I'm going to leave number 50 for you guys to answer, and I'm going to skip ahead with 51. Tutut! Mabilis. Grabe, sir, ang bilis. Hindi ko nakita. Di ba? Ang bilis. Nakita nyo ba? Nakita nyo? Mabilis. Masyado. Sobrang bilis. Oh, somebody answered already. Deandra Postigo. Okay, she says 50, ano tong 50 Gcash? Magbigay ako ng 50
Hindi po yan yung number.
Okay. Ay, oo, yung ano.
Or baka sabihin nila mo akong pera. Hindi po yan,
Girl, wag kang mag-alala. Pag mukhang pera, I love it. Kasi ako, mukha din akong pera, girl. Wag kang mag-alala. Bakit nga ba ako nagtatrabaho kung hindi ako mukhang pera? Wala akong pang-load sa Genshin Impact ko. Ano ba? Kayo talaga? Siyempre, kailangan mukha tayong pera. Di ba? Okay? So, Aperitricus flagella is observed in all species of Enterobacteria C, except for which one? We have several, I give you choices na. Isa-isahin natin from the very start of what we're going to discuss. Okay? You can use the chat box, guys. You can use the chat box. I'll give you guys choices. You can only select one. This is the only one that does not have all Peritricus flagella can be observed in all species of Enterobacteria C except for which one? These are your choices. Escherichia coli, Enterobacter, Klebsiella, Pseudomonas, Edwardsia, what else is there? Labibacterium, Plesiomonas, Helicobacter, what else? Vibrio cholerae, Salmonella. O, yan na. Madami na akong binigay na choices. Aperitricus flagella is observed in all species of Enterobacteria C except for those 10 that I just said. It's one of those organisms in the choices. E. coli, Enterobacter, Klebsiella, Salmonella, Pseudomonas, Labibacterium. Ano pa ba? Wala na akong maisip eh. Basta nandun sa first 6. Basta nandun sa first 6. Sige na. Sige, pwede kayong manghula. Huwag kayong mag-alala. Pwede kayong manghula. Hindi ko kayo i-judge. Si Sir Marco, baka i-judge kayo. Pero ako hindi ko kayo i-judge. Kasi alam ko limited ang resources natin para mag-aral this time. So don't worry. I will not judge. Okay, mahiya. Di ba, Deandra? Pwede nyo ding ilagay 51. Malay nyo, magbigay ako ng 51 Gcash load. Diba? So type in your answers for this one. We're going to skip. Oh, somebody answered already. So, hindi na ako nag-skip. Thank you so much. So, Marivic is actually correct. Her answer is correct. It's Klebsiella. So, Marivic, I want you to unmute yourself because you answered Klebsiella. Or Klebsiella. Sorry, Klebsiella tuloy. Klebsiella. Okay? Marivic, where are you? I need to talk to you, girl. We need to talk about something very important. Charing. Kala mo interview. Okay. So, what is the location of the flagella? What is the flagellar structure of Klebsiella? Is it atricus, monotricus, the other trichuses? Is it periaxial? Does it have periaxial filaments or the other ones? Monotricus, lopotricus, atricus. What is it?
Hindi ko po sure.
Sige lang, hula ka. No problem. The other people can also guess. So you guys can help out Marivic. Okay. Sige, you can guess. Don't worry. In this class, we are not here to judge. We are here to learn. Kasi pag nagkakamali kayo, at least naaalala nyo, di ba? Somebody is correcting it. Di ba? Sige, you can guess. Bigyan kita ng clue. IMVIC result niya is negative, negative, positive, positive. Doon pa lang makukuha mo na, oh my God, oo nga sir, no? Alam ko na dapat ang sagot. Diba? Kung ang IMVIC result niya. Kung alam mo ang ibig sabihin ng IMVIC. Diba? Grabe, ang dami ko na binibigay na ano sa inyo, ha? Ang dami ko na binibigay na clues sa inyo. Kasi yung Klebsiella, ano siya? Kung negative, negative, positive, positive siya. Diba? Oh. We're going beyond general discussions. We're going to the specifics of an organism. But I think this needs to take precedence over the basics. Because if you do not understand this, when we go to the exams or when we go to the specifics, you may not be able to answer the questions. I need you guys to let these types of knowledge or information that I'm giving to you marinate in your noggins. So, E. coli. Hoy, sino nagsabi ng E. coli? Ang tinatanong ko, ano na ang structure niya? Monotricous? So, the correct answer is pronotricous. Very good. Arnie Salan. Arnie Salan, I have not seen your face. Where are you? Ay, ayan pala siya. Hi, Arnie. I love the Jose Rizal hairstyle. Actually, that's my hairstyle yesterday. I love it. Okay, so why did you say it's atricus? Looking at the IMVIC results, why do you say it's atricus? Because it tested what? Yes, I can tell what you're saying, and you don't need to say the words. We have a psychic connection. Yes, very good, Arnie. Yes, you said that because it tested negative for sulfide indole motility. It's negative, di ba? Ano ba yung M? Ano ba ang agar niyan? SIM, SIM. Di ba? SIM agar, sulfide indole motility. So if it tested negative for IMVIC, obviously this is an organism that will not move. Obviously, this is an organism that does not create an indole reaction, and therefore there is no flagella. 'Di ba? 'Di ba? Ang daling i-connect, 'di ba? Kapag meron kayong baseline understanding of everything. 'Di ba? Okay ba 'yon? Bet mo 'yon, Valiant. 'Di ba? I am serving you well. She is the moment. She did everything that needed to be done. Ganon, 'di ba? Okay, next. So, except for Klebsiella, all right? Klebsiella species. So, what type of flagella is observed in Vibrio species? Ayan na. Vibrio naman. Same choices, atricus, monotricus, lopotricus, blah, 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 tricus. You guys have several choices. Okay? And again, I have another discussion on that one. That's the reason why I want you guys to know. Alright, so last na nakasagot is si Arnie. So therefore, the next person who would answer would be my baseline. So I'll write this so I know. Sorry, oops. What happened? Oh. I'll write this here so I would know who answered who answered. Okay, so what's our answer for this one? Anybody? Ito na ang choices, baka nakalimutan. Ibabalik ko, baka nakalimutan ang choices. I'm sorry. I apologize. Okay, so those are your choices. So for Vibrio, what is the structure or the flagellar structure or morphology? Is it atricus, monotricus, amphitricus, lopotricus, peritricus, or periplasmic flagella or axial filaments? Monotricus is incorrect. I'm sorry, James Paul. Oh, sorry. You are correct pala. Sorry. Monotricus, sorry. You are correct. Okay, so yeah, James Paul is correct.
Muntik na ako mag-react.
Pero hindi sila sure. Ayoko nang hindi sure. Di ba, Sir Marco? Sorry, mali pala. Mali ako mali. I agree with James Paul. It's monotricus. Ganon. Parang walang conviction. Monotricus. Parang ganon. I don't know, but I'm not sure. But I think it's monotricus. Yeah. So, bakit? Ano bang motility pattern niya? Falling leaf, darting motility, sluggish motility, or no motility at all? Sige. Okay lang yon. Huwag ka mag-alala, James Paul. Ano ba? Anong klaseng motility meron siya? Tingnan natin in terms of parasites. 'Di ba madaming flagella si ano? So parang kakaiba yung itsura niya. Yung isa naman sluggish kasi nga madami din. Eh siya may isa lang, monotricous lang. So therefore, ano ang movement niya? 'Di ba? Ganun. Anong magandang example ba?
Corkscrew, sir.
Corkscrew? Pwede din. Sige, corkscrew. Sinabi ko na nga yung darting motility kanina. Ganun na din yun. Corkscrew. Sige, sure. Alright, very good. But corkscrew actually works more for Helicobacter pylori, but I would accept it for you because sometimes there are some books that says it's actually corkscrew. But for majority of the references that I've used, it's darting motility. But okay. All right. Now, examples of tests for motility, you could use a hanging drop method. You could use staining the flagella, or you could use a semi-solid media. Ano yung semi-solid media, sir? SIM, sulfide indole motility agar, or the SIM agar. Okay? Now, let's discuss the motility of other organisms. Okay? So, species belonging to Campylobacter, they have darting motility, as I mentioned before, because they're almost the same as Vibrio. Okay? The only difference between them is that their infection rates is different, or they're quiet, but they're genetically similar to each other. So there are just some insertions that are different. So gliding motility. This is also a monotricous organism. Capnocytophaga is another one that you need to remember. This is an organism that is part of the Hasek group. Okay? So you guys need to remember Hasek group also. Capnocytophaga is one of them. Okay? Echinella corrodens. Let's go from Capnocytophaga, Echinella corrodens, Cardiobacterium hominis, Acinetobacter or Actinobacter. It can be in the same group. And then Hasek H is your Haemophilus. Okay, so don't forget. What is the Hasek group, by the way, guys? These are organisms that are what? That are anong tawag dun sa maarte? Sige, anong tawag dun sa mga maarte na organisms? They are fastidious organisms. When I say fastidious, these are microorganisms that always go to the Starbucks. Okay? They want Starbucks coffees and in the form of CO2, right? They don't like oxygen. They don't like the absence of oxygen. They also don't like microaerophilic environment. They like CO2. Okay? So the Hasek group. So Echinella corrodens, Capnocytophaga, and those kinds of things. Alright? Now, Listeria monocytogenes grows on what media? Okay? Listeria monocytogenes grows on sorry, ano ba? Sorry, naging lalaki. Tumbling motility on McBride medium. Okay? It has a tumbling motility. Capnocytophaga is more on gliding. How do I characterize gliding? Can you guys see my mouse? Or my laser pointer? Can you guys see my laser
Hindi
pointer?
po. Ayan, sir.
Nakita nyo na,
Ayan
sa
po.
gitna, diba? So, how do I characterize gliding motility? Darting motility. So, gliding motility is parang, wee, ganun lang. Okay, dahan-dahan lang. Si darting motility, ganun, Pero hindi ganyan kalaki ha. Parang slow, ganun siya. Okay? So it's very demonstrable or extremely observable when you're doing hanging drop preparations. Especially for these two organisms. Okay? All right. Next. What is the most common type of media used for bacterial motility? Ayan na, sinabi ko na. Semi-solid media used for using SIM. Okay, sulfide indole motility media or agar. Sulfide indole motility agar. Okay? So what does it look like, guys? Do you know what it look like? What it looks like? Is it in a plate or is it in a tube? Sige nga. Is it in a plate or is it in a tube? Write your guesses while I Google search it for you guys. Okay, write your answers. Is it in a plate or is it in a tube? Or if you want me to be more specific, kasi gusto ko nga naman ng specificity, tapos hindi ako nagpa-practice ng specificity. Is it on a plate or a petri dish or on a tube? Ayan. O diba? Meron akong picture para sa inyo. This is from University of Wyoming. Ano namin nag-guess? Tube. Okay, so people answered tube. Walang magsasagot ng nasa plate. Walang nangangahas. Kasi parang tama yung nasa tube. Or parang hindi tama yung nasa tube. Walang nangangahas. No? Talagang sila, Sir Marco, talagang sila, tag doon? Standing together. Diba?
Siyempre, para pag mali lahat.
Oo. Ayan ang itsura niya, guys. Alright, ganyan yung itsura. Nakikita niyo? Okay?
Yes po.
This is credits from the University of Wyoming. So, YouTube please don't demonetize this video. Charot! Ganyan na talaga, no? Alright, so how do you use SIM? What's the plating, or what you call the inoculation technique, rather? How do we inoculate your organism there? Ay, ano yung dinidemo mo, Mark? Ano yung dinidemo mo?
Inaalala ko lang po kung paano.
Anong tawag doon? We need specific terminologies. We're doing science, remember? We need specific terminologies. O, ito na. Sasabihin ko na sa inyo. Okay? Kasi alam nyo naman, kahit ngayon lang tayo nagkita, mahal ko naman kayo. Huwag kayong mag-alala, guys. 'Di ba? Stab. Okay, people said stab. Alright, James Paul. Thank you so much. Hindi ba pwedeng stab and streak, James Paul? Sige, tatanungin ko muna si James Paul. James Paul, where are you? Where art thou, James Paul? Parang ang tagal nating hindi nagkita. Okay, James, bakit hindi pwedeng stab and streak? Sige.
Stab lang po, sir.
Bakit ga? Bakit stab lang? Ikaw ang istabin mo ng inoculating loop dyan.
Ano lang po kasi.
Okay, so tama naman ang sagot niya. Stab lang, pero incomplete. Who knows, who can complete the answer for me? Alexis, sige nga.
Yung idea ko po kasi, sir, for stab and streak po is for butt slant medium lang po siya, sir.
Okay. Very good. Okay. Any other answers? Parang mali ang sagot ni Alexis. Char lang. Tama naman, kinda, but it's a little bit incomplete. Okay. Sige, ayun na si Nigel. Sige. Parang galit si Nigel.
Tampo.
Tama naman sinabi ng mga kaklase ko, ha. Sige, go.
Stab po kasi na-observe mo yung motility ng bacteria.
And? Okay, so ino-observe natin ang motility. So, what is the reasoning behind that one? You are only stabbing. Yeah, you're correct. You're also correct. But again, the details. I'm looking for details. Why only stab? Alright, so, siret na. Partially correct lang kasi yung mga sagot. Hindi ako papayag na partially correct. Gusto ko complete correctness. Charot! O, yan na, Nigel. Nag-raise ka na naman ng hand mo. Sige, go. Ay, hindi na.
Ito lang po nababa.
Okay. So, wala nang gusto mga has na mag-try. Okay, sige. So, I'll answer it for you guys. Kasi, guys, may solid siya. the agar will close on itself. Diba? Diba? It's semi-solid. Kanina ko pa sinasabi kaya I put emphasis on semi-solid. Kasi if it's bordering the realms of what? Of a solid medium and a liquid medium. So, what you need to do is you just need to make an impression. 'Di ba? So, kaya ka mag-stab lang. You just need to make an impression. It will return on itself, but it will still make an impression. And if the organism moves in that small piece of space in between, it is going to spread out. And therefore, you are observing. As Nigel have said, observe lang natin yung motility, sir. 'Di ba? Tama. You're correct, but partially correct. Okay? Now, I'm going to ask you guys, what did we add here? On the second tube. Why is it color red? Sulfide indole motility. A SIM media will never be complete, or a test for SIM or test for motility will never be complete without this one. My first question is, why is it red? Ayan, o diba? Grabe naman to, sir. Ang hirap. Ayoko na mag-medtech. Gusto ko na lang mag-drag queen. Woo! Gusto ko na lang maging singer. Mag-DJ na lang ako. Can this be love? Ganon. Gusto ko na lang maging heartthrob. Ganon. Ano? Bakit color red siya? Why is it red? It's the same agar. Right? The same agar. So what made it red? Look at the reagent, oh. We all added a reagent on it. So what's that reagent that I added? Makasagot niyan sa akin. Makakasagot niyan sa akin. O sige, may nagtaas ng kamay. Sige, Randolph. Randolph, this is your first time. You introduce yourself, Randolph. Okay? Where are you, Randolph? Where is Randolph?
Hello po, sir.
Oy, oh my God, nakakatakot
Hello po.
yung voice. Oh my God, he's very dreamy. The voice is very... I need to see your whole face because this is important. Okay, oh my gosh, I love also the, what do you call this? The Jose Rizal hair. I think it's trending in the Philippines. I love it. Okay, so what's the reagent that we use here? You can
Sir,
use the
sa
acronym.
simple reagent po. Sir, di ko po, sure.
Oh sige, bakit ka nag-raise ng kamay? Hindi ka pala sure.
Ay, hindi, sir. Kakapasok ko lang po. Nag-CR lang po kasi ako.
Okay.
Sorry po.
Visit
Kakabalik
ka,
ko lang.
Randolph. Akala ko
My
naman,
bad po.
alam mo, napakamalam. Akala ko naman, malaman yung sasabihin mo, Randolph. Kainis
Sorry
ka.
po, sir. Bawi
Anyway,
na lang po
sige,
ako.
who else knows? Parang alam ni Alexis. Alexis. Ay, may nag-raise ng hand. Si Alexis. Alexis, sorry. May nauna sa'yo. Si McKay. I'm calling her McKay. Okay. Ano na? Bakit nag-raise ng hand? Hindi nagsasalita? Ano ba?
Ay, nakamute pala, sir.
Ayan na,
Indicative of positive indole po.
Alright. Okay. Oh, what reagent did we use naman? So, okay, indicative of what? You can't get this reaction without adding a reagent, and that reagent is important. What is that one? James Paul, we would like to help you. Would you be okay if we helped you? Ayaw niya.
Sir,
James,
muna?
ayaw niya, James. Sorry.
Sir?
O sige.
Guess
Fine.
ko muna. Covax po. Covax
No, Kovacs
reagent.
reagent is different. That is used for a different test altogether. Pwede nyo gamitin yung acronym. Hindi ko kayo pahihirapan. Acronym siya. Ayan, hindi na makatulog si James. Nakakatina si James. Sige, nakatawagin ko na si James. James, sige na. Go.
Sir, di ko sure, pero hydrochloric acid po,
Hoy,
tsaka
pinatay mo yung mga
p-dimethyl.
mikrobyo. Hoy, pinatay mo yung
Sir,
mga
ano po?
mikrobyo. Nasunog ang agar.
Tapos ano po, p-dimethyl aminobenzoate.
Ayun! Akala ko naman, incomplete na naman eh. Very good. Ulitin, pakiulit for the class. Pakiulit. Medyo naglag eh.
P-dimethyl aminobenzoate.
Plus,
Tsaka hydrochloric acid.
very good. Padma. Okay? Padma or para-amino dimethylbenzaldehyde. Okay? It's para-amino dimethylbenzaldehyde. producing that. Actually, it's supposed to be pinkish. It's supposed to be pinkish. It's not supposed to be reddish. I don't know why it's red, but it's supposed to be pink. But whatever. That's fine with me. Now, next is the other tube. Why is it black? Oh, that's easy na. That's easy to answer na. You don't need to add a reagent. Oh, yan na. Babalik tayo. Sulfide indole motility. Ayan na, nakakainis kayo pag hindi nyo nasagot yan. Magigigil kami ni Sir Marco. Ano na yan black? Bakit naman yan black? Ano ba yan, sir? Hindi ba pwedeng bowtie lang? Naiinis na ako sa inyo. Magda-drag queen na lang talaga ako. Magsi-singer na lang ako. Sasali na lang ako sa tawag ng tanghalan. Okay, bakit siya black? Ito isa, bakit black? Who can answer? This one. Tama ba yung pinupoint ko? Tama ba pinupoint ko, Mark? Tama ba to?
Guys, tatlo lang yung test. It's
Yes, po.
literal sulfide indole motility.
Kaya nga eh.
So, elimination na lang yan.
Nagigigil ako sa kanila talaga, Sir Marco eh. Kinalimutan ang basics na. Kanina ka mas masagot mga advanced na mga sagot. Arcanobacterium eh. O sige. Sino ba nauna? Sandali, sino nauna? Dali, bago ako baka sabihin unfair ako. Sabihin baka dahil lalaki si ano. O sige na, si ano muna kay Maria, kay McKay tayo. Hindi, McKay.
Na-produce po yung hydrogen sulfide.
Very good. That's easy. See? Wonderful. Bigyan ng jacket. Kaso walang plus eh. Ay, walang ano eh. Kasi very basic. Magagalit sa akin si Sir Marco pag nabigay ako ng pera niya ng napakadali na question. Ayaw niya, gusto niya yung sobrang hirap. Okay? So differentiate the pathogenicity from virulence. This one is a different topic altogether. We're talking about the pathogenic determinants. Yes, sir.
Sir, I have a question lang. Kasi I think one student mentioned COVAX kanina.
Yeah.
Component kasi ng COVAX is para-aminobenzaldehyde. So, correct me na lang if I'm wrong.
Oh, yeah. Sorry. I forgot to talk about that one earlier. Yeah. He did mention about I'm not going to remove that. I'm not going to take that away from him, but I'm actually looking for the actual reagent, the actual chemical. So, I forgot to mention
Okay,
about
baka
that.
lang kasi maisip nila na COVAX is wrong.
Yeah. I forgot to. I think James Paul was the one who said that. I'm terribly sorry, James Paul, if I made it sound like your answer was not valid. But yeah, you are correct. But what I was asking for is actually the reagents, the contents of the reagents. Like I told you guys before, you can give me the complete name, or you can give me the acronym. Okay, so COVAX reagent is actually used for indole demonstration, and the components of it is, as he said, it's para-aminol dimethylbenzaldehyde, and it contains hydrogen chloride. Oh yeah, hydrogen chloride, or hydrochloric acid rather, and the reason why I didn't accept COVAX reagent is because you can use COVAX reagent in another test. Okay? So this is just one of the tests where you can use a COVAX reagent, and I think it does make it's important to give merit to somebody who said something that is correct. And I forgot to mention about that. Parang hindi ko ata nadaanan ko lang ata kanina. Buti pinaalala mo. Thank you, Sir Marco. Love you lots. Hugs, kisses. Now, can we move on to the next one? Okay. So we're going to talk about the next one. Okay. So the next one that we're going to talk about is pathogenicity. So we're going to talk about the pathophysiology of microorganisms. So we moved on from physiology, and now we're moved on to the pathogenesis of how microorganisms are capable of causing diseases. Okay? Now, can somebody differentiate to me what's the difference between pathogenicity and virulence? Sige. You can say it in Tagalog, but because you guys are beginners, okay? So I'm not going to expect that much from you. What's the difference between pathogenicity and virulence? They are related to one another, but they are different. They have different definitions. Sige. I think it's important that we give them a bit of a break. 15 minutes is enough, because my tummy is rumbling, and my tummy is rumbling, and I think I'm hungry. So because it's already 12, I think it's already lunchtime here in my location. So I'm leaving it to you guys on the chat box. What differentiate? Oh yeah. I'm not leaving it in the chat box. I want you guys to answer the question. Number 56. Okay? Number 56. Please answer. Okay, para alam ko kung kailan ako, anong babalikan ko. Okay? So yun. Okay. 56. Please answer. Okay? The difference
Yes
between
po.
pathogenicity and virulence. So we're going to stop there. I'm also going to stop the discussion in the live stream on YouTube. And I hope you guys will have a wonderful break time. You guys have been wonderful. The link for our live stream is going to close. Please make sure that it's actually closed. Mark, can you help me out?
Yes po.